días. Buenos días a todos quienes asisten al primer webinar internacional de Story of Microorganisms in the Context of International Microorganisms Day, organizado por el Centro de Investigación y Estudios Avanzados.
Alex, su micrófono.
Sí. Pero todavía no empieza el Zoom.
Estamos en vivo.
Muy buenos días a todos, a todos quienes asisten al primer webinar internacional titulado The Story of Microorganisms in the Context of International Microorganisms Day, organizado por el Centro de Investigación y Estudios Avanzados del Maule, perteneciente a la Universidad Católica del Maule de la ciudad de Talca, en Chile. Agradecemos su presencia en lo que será una doble jornada que contará con la participación de destacados científicos de nivel internacional. Para comenzar esta primera jornada, se dirigirá a ustedes con un saludo la doctora María Teresa Muñoz Quesada, vicerrectora de Investigación y Posgrado de la Universidad Católica del Maule.
Estimadas autoridades, investigadores invitados, académicos y estudiantes, a nombre de la Vicerrectoría de Investigación y Posgrado de la Universidad Católica del Maule, les doy la más cordial bienvenida al webinar internacional La historia de microorganismos en el contexto del Día Internacional del Microorganismo, actividad organizada por el Centro de Investigación en Estudios Avanzados del Maule de la Vicerrectoría de Investigación y Posgrado y el Centro de Biotecnología de los Recursos Naturales, CENBio, de la Facultad de Ciencias Agrarias y Forestales de la Universidad Católica del Maule. Como vicerrectoría, buscamos contribuir a mejorar la calidad de vida de todos los chilenos mediante la generación y aplicación del conocimiento científico y tecnológico emanado desde la Universidad Católica del Maule, velando por la solidaridad, la ética y los principios de la fe cristiana. Nosotros definimos 3 áreas de investigación prioritarias: medio ambiente, recursos naturales y energía, sociedad, educación y familia, y salud y calidad de vida. Dentro de la primera, tenemos 2 subáreas relacionadas con la biodiversidad: biotecnología y agroindustria. Esta es una de las áreas más productivas de nuestra institución, ya que se vincula fuertemente con los recursos naturales y la biodiversidad ecosistémica que presenta nuestro país y, por sobre todas las cosas, nuestra región del Maule, y la especialización de nuestros investigadores e investigadoras responden de forma efectiva en esta necesidad. En esta sublínea nosotros no solamente apoyamos los procesos de desarrollo de cada proyecto de investigación, sino que también difundimos y queremos transferir estos conocimientos al servicio de la comunidad científica y la comunidad en general, con el fin de aportar en la mejora de la calidad de vida y el bienestar de las personas, siempre respetuosos en la conformación y la cultura de los territorios por la vida de todo ser humano y cuidado del medio ambiente. Es importante reconocer también la colaboración y el trabajo en equipo que desarrollan los investigadores e investigadoras del Centro de Biotecnología, del CENBio, y con el Centro de Investigación de Estudios Avanzados del CIEAM, que ha permitido generar resultados de investigación en conjunto con impacto para velar por la conservación de los recursos naturales y no solo adjudicar proyectos relevantes de investigación y potenciar la difusión de los resultados en revistas indexadas de los primeros cuartiles, sino que también impactar positivamente en la región, en el sector agrícola y forestal, en la formación de pre y posgrado y en la comunidad en general por la promoción de un desarrollo más sustentable. Este webinar apunta hacia esa visión. Además de compartir nuestras experiencias internacionalmente en el área de la microbiología, creemos que valorar la preservación del patrimonio microbiano e incentivar la investigación científica y aplicación de esta área en los nuevos desafíos que se nos vienen como especie, de la búsqueda de tecnologías limpias y que velen por el bienestar del ecosistema natural en su globalidad, permitirá en cierta medida dar respuesta a la crisis climática que estamos viviendo y en la cual hemos sido responsables como humanidad. Esto que menciono se refleja en las temáticas y actividades que se van a realizar en el webinar y que será un espacio participativo de investigadores de diversas naciones y con destacada trayectoria en sus disciplinas y líneas de investigación. Finalmente, quiero agradecer al CENBio y al CIEAM y a los invitados nacionales e internacionales que nos compartirán su experiencia en investigación y sugerencias, y esperamos seguir potenciando este vínculo con proyectos e ideas en conjunto que contribuyan en las temáticas de la protección de la diversidad microbiana a nivel mundial. Espero que disfruten esta experiencia y sea un espacio constructivo, participativo y de aprendizaje significativo para todos ustedes. Muchas gracias.
Thank you so much, Dr. Maria Teresa, our Vice Rector from Universidad Católica del Maule, for your welcome talk to the international webinar, The Story of Microorganisms in the context of International Microorganism Day. From the part of Universidad Católica del Maule, I welcome you all to today's international event. So now, with a continuation to our Vice Rector's welcome talk, I will present to you the welcome talk of our Director of Research, Dr. Ingrid Carvacho Contreras from Universidad Católica del Maule.
Good morning, everyone. On behalf of the Direction of Research and also as Director of Center of Research and Advanced Studies of Maule, Centro de Investigaciones y Estudios Avanzados del Maule, of Universidad Católica del Maule, I welcome you all to our international webinar, The Story of Microorganisms in the Context as International Microorganism Days, organized by Dr. Aparna Banerjee, Assistant Professor at our university. This event celebrates the International Day of Microorganisms, which was on the 17th of September, but because of the closest to our national day, that was the 18th, we decided to celebrate it today and tomorrow. And the main goal is to promote the diversity and variety of microscopic life, and of course, to celebrate the importance of them in our daily life. As a biochemist, I'm very happy to welcoming you all to this important event that will be running for 2 days, and during that time, we will enjoy talks from experts from USA, Mexico, Spain, Chile, and India. We will discuss about extremophile and infectious bacteria and bacteria derivative bioactive compounds and much more. The interesting thing to have this webinar in pandemic time is that actually the pandemic gave us the opportunity to develop a new way of communication where the physical distance is not a problem, as you can see in this organization. As Universidad Católica del Maule, we are very happy to be part of this, and we hope that will be a great experience for you, for all of you. We invite you to enjoy from home, to develop ideas, to better understand the power of the microorganism. And finally, we invite you to generate a fruitful discussion that may open new avenue of research and collaboration of our university nationally and internationally in the field of microbiology. Thank you very much for to be here and enjoy the meeting.
Thank you so much, Dr. Ingrid Carvacho Contreras, for your welcome talk to our webinar. So with continuation, the first presentation of today's event will be of Dr. Rajender Deora. Dr. Deora completed his master's in biochemistry from University of Kolkata, India, and PhD from University of Illinois at Chicago. After completing his postdoctoral studies at University of California at Los Angeles, he joined as an assistant professor at the Department of Microbiology and Immunology at Wake Forest University. He's currently working as associate professor at the Ohio State University, United States. So Dr. Deora, please.
Hang on just a second. Can you hear me?
Yes.
Let me share the right screen. Okay. Well, thank you, Dr. Aparna Banerjee and University of Católica del Maule for this invitation. I'm really happy to present our research, and like it was said that the pandemic gives us an opportunity to have people from many countries, and I think this is the first time I'm giving a talk where so many people from different countries and different time zones are attending. So what I'm going to do today is talk to you about one particular pathogenesis factor or the virulence factor, that is BPS. Yeah.
Dr. Deva, can you please put it on full screen?
Okay, hang on. I thought it was on full screen. It is on full screen, at
But
least on.
Mm,
Is it better now?
No, if you can just put this one, yes.
Is it better?
Actually, it's not putting full screen even if you click. So I think it's okay. You can continue. It's okay. You can continue.
Let me stop share and then start, see
Okay.
if I can
Okay.
start
Okay.
again. Let's see. Is it better? A little bit better now?
It's not yet there.
Okay. What can you see? Can you see the slides?
No, now no. You have to go into the presentation mode again, probably.
Okay, I might have to share the screen again. Hang on. Is this better?
It's okay. Now it's okay. So let's start.
Okay.
Now.
Okay, so we are going to talk about this virulence factor, the BPS polysaccharide of Bordetella pertussis. So Bordetella pertussis is, hang on, let me. Can you see my cursor?
Yes.
Okay. Bordetella pertussis is an obligate human pathogen and is a gram-negative bacteria that colonizes and attaches to the ciliated respiratory epithelium of human hosts. As like I said, it's an obligate human pathogen that means that it does not have any environmental reservoir or any animal reservoir. And remember this property of Bordetella pertussis, that it attaches exclusively to the ciliated epithelium, which will become important in the later part of my talk. So Bordetella pertussis is associated and is the causative agent of whooping cough or pertussis. This disease is very severe in unvaccinated or incompletely vaccinated infants, but in the last few decades, there have been more and more cases of pertussis being detected in older children and adults who are fully vaccinated. So before I go, let me try to show you a video of that. This is a 2-year-old girl who was diagnosed with pertussis. And I hope all of you could hear the sound of the baby girl and what she's trying to breathe but cannot breathe, and she's forcing herself to breathe, and that's why you can hear the whoop in between the breaths, and that's the typical name of this disease, whooping cough. So pertussis, there are a number of vaccines available for pertussis. The whole cell pertussis vaccine, also known as WPV or DTP, was introduced in the 1940s. It is still used in many countries, but in a lot of countries, the European countries and in USA, because of the reactogenicity, severe reactogenicity like pain, redness, swelling, and fever, they switched to the acellular pertussis vaccine. That means the acellular pertussis vaccine does not have any bacteria, but it has purified components of Bordetella pertussis. And there are different versions of the acellular pertussis vaccine for young children, for preteens, for pregnant women. In USA, they have started vaccinating pregnant women in the 27 to 36 week of pregnancy. And then there is a version for adults. So when you think about it, with so many different options available, wholesale and acellular, you would think that the pertussis would be well controlled. But the reality is, because of the switch to acellular pertussis vaccine, even in countries which have wholesale pertussis vaccine, pertussis is re-emerging. So in 2018, WHO estimated 151,000 cases of pertussis globally and 89,000 deaths. And this is in the background of pertussis is very difficult to diagnose in patients. And so another study estimated about 24 million pertussis cases and 160,000 deaths. And I am not surprised because a lot of times people even don't, physicians don't even diagnose for pertussis. So I thought I was able to pull out some data from Chile, and in Chile actually switched in 2018 from the DTP to the acellular pertussis vaccine. And I was able to find this from a website where Chile reported actually very high number of cases of pertussis or whooping cough. And until December 13, 2020, there were 4,277 cases and 9 deaths. And this is kind of striking because in 2018, they switched to the acellular pertussis vaccine. And in 2020, we are seeing an increase in cases. And I really hope, keeping my fingers crossed, that these numbers don't keep increasing like what happened in other countries when they switched to acellular pertussis vaccine. So the vaccine efficacy of the acellular pertussis vaccine is not very good. What has been found that the acellular pertussis vaccine, although it prevents the severe symptoms, that is the whooping cough that you heard in the baby girl, this acellular pertussis vaccine does not prevent the colonization of the vaccinated individuals in the nasopharynx with the bacteria. These vaccinated individuals still carry the bacteria in the nasopharynx. And what they end up doing is they end up transmitting these bacteria to susceptible or partially or unvaccinated infants and younger children. And this keeps on continuing the circulation of the bacteria. So that's what we are interested in. We are not only interested in development of vaccines for this, better vaccines for this bacteria, but to understand the basic mechanisms of pathogenesis. So like any other bacteria, Bordetella pertussis goes through a number of steps. The bacteria gets transmitted and it infects new hosts. And once it infects, it initiates colonization in the nose, trachea, and the lungs. And there are two outcomes of this colonization. One is asymptomatic infection, that is the bacteria are carried in the nasopharynx without any overt signs of disease, or they end up causing disease. Again, I want to emphasize that the disease is mainly in unvaccinated or partially vaccinated individuals. So our hypothesis for this is that Bordetella pertussis ends up forming biofilms in the nasopharynx of the vaccinated individuals, which results in better colonization and asymptomatic infection. And ultimately, the bacteria gets transmitted to susceptible individuals. So we started studying biofilms of this bacteria. So I know that there are a lot of undergraduates who are in the audience, so I thought I would just give a brief description of the biofilms. Biofilms are defined as communities of surface-bound organisms which are encased in a self-produced, either produced by the bacteria themselves, and or they could be an host-derived polymeric matrix. So a lot of times during infection, the biofilms are not only have bacterial matrix components, but also host-derived components like from the neutrophils, nets, and other DNA from the host. So like a lot of the labs studying biofilms, we initially exclusively studied biofilms on abiotic surfaces for, like you can see on the glass, Bordetella pertussis form really robust biofilms. This is GFP tagged bacteria. We can see biofilms as a form of a ring when we grow bacteria in a roller drum. By scanning electron microscopy, we can see the biofilms, and this white substance is what is called the matrix of the bacterial biofilms. So we actually have characterized and shown that biofilm matrix components are composed of eDNA, extracellular DNA proteins, and the BPS polysaccharide. And we have shown by taking mutants or enzymes that all three components are required for integrity of the biofilms. But today, as I said, I'm going to focus on the role of BPS polysaccharide in pathogenesis and biofilm formation. So what is BPS polysaccharide? The BPS polysaccharide is believed to have a structure of polymeric beta-1,6 linked and acetyl glucosamine. So when I say beta-1,6, this is a very unusual linkage in carbohydrates where the monomers are linked by a beta-1,6 linkage. And this review actually described the role of different polysaccharides in biofilm formation in pathogenesis. And here is the PNAC polysaccharide, or the polybeta 1,6-linked N-acetyl glucosamine polysaccharide. And this is a large family found in diverse bacteria. This is only focused on the gram-negative bacteria, but this is also found in gram-positive bacteria, especially in Staph aureus, where PNAC polysaccharide, and in E. coli, it's a PGA polysaccharide. And in case of Bordetella bronchoseptica and Bordetella pertussis, this is the BPS polysaccharide. So after having studied the in vitro biofilms for a long time, it kept on bugging me, is that what do we actually learn from in vitro biofilms, and what is the therapeutic significance of in vitro biofilm? Can you really devise therapeutic based on solely the studies of in vitro biofilms? So what we started asking is, can we detect and study biofilms in mammalian hosts? So for Bordetella pertussis, the field uses generally two models largely for mouse infection. One is the intranasal model, where you inoculate bacteria through the two nasal cavities of mice. And another is the aerosol model, where you create bacteria as an aerosol, and then the mice end up inhaling. And as soon as you inoculate bacteria, there's initially a lag period, and then the Bordetella pertussis very quickly replicates and colonizes the trachea, nose, and the lungs. And it stays in the mouse, all three organs of the mouse, for a pretty long time, about 35 to 40 days post-inoculation, and then these sites are cleared. So we decided to use this mouse model to see, first question was whether Bordetella pertussis forms biofilms. So what we did was we infected mice with Bordetella pertussis and then took out the nasal septum and the trachea. And I want to emphasize that these organs have not been manipulated in any way except to stain them. The mouse epithelium was stained red, where you see here red, and the bacteria are green. And the bacteria was stained with an antibody specific for Bordetella species. And what you see is that one day post-inoculation, immediately after inoculation, Bordetella pertussis has started forming this community-like structure on the nose and the trachea. And 7 days post-inoculation, I don't even have to tell you that these are biofilms, where you can see these very large communities of bacteria spread along the epithelium. And you have to remember, Bordetella pertussis is a small microorganism, about 0.3 to 0.4 microns, and this is a 5 micrometer bar. And when you consider the thickness, you can appreciate the layers and layers of bacteria that are forming on the host epithelium. So we also used an independent method, scanning electron microscopy, where you can see the bacteria are colonizing the cilia of the present in the nasal septum. So this is again what is typical of Bordetella pertussis, where they exclusively colonize the ciliated epithelium. So the next we asked is, what are the determinants of biofilm formation on mouse respiratory tract? So what we did first is we compared the wild type strain with the mutants. This is FHAB, is a mutant in the Bordetella protein, which is also an adhesin for Bordetella, and it is included in the current acellular pertussis vaccine. And what you see when you infect the mice with the mutant strain forms very thin spikes, and essentially it is devoid of biofilms, but it's not completely, the bacteria are still there. It's just that the biofilms are sparse. We next tested the Delta BPS AD mutant, and to our surprise, we found that the Delta BPS mutant was not detected at all on the nasal septum of mice, suggesting that it is involved in biofilm formation and probably in the colonization of the mouse nose. We also, what we did was we tested the role of DNA. We took the nasal septum out and treated with the DNase buffer or DNase. And what we saw that ex vivo treatment resulted in the complete disruption of the biofilms formed on the nasal septum, suggesting that DNA is required for the integrity of the biofilms. So then, after having studied the mouse biofilms for quite a while, it kept on bugging us that mice are not humans. It's a really good system to study in a mammalian host, but given that Bordetella pertussis is an obligate human pathogen with no environmental or animal reservoir, there is only so much you can learn from a mouse. So what we decided was we thought that to precisely understand Bordetella pertussis pathogenesis, we need to develop more human relevant model systems. So what we decided was, before I go in there, is there an evidence for Bordetella pertussis biofilms in humans? So this is a picture taken from a paper published in 1912, where in those days they did not even take pictures. These were drawings by the authors, and what they reported was from the lungs from patients who died of whooping cough, they detected masses of minute bacilli packed between the cilia of the lining epithelium and in the secretion in the trachea and bronchi. And even though biofilms were not known at that time, but these masses of bacteria, which is designated by here, suggest that these could be the biofilms. And then in 2008, Paddock et al. Reported aggregates and tangles during fatal infection in infants. I think this is a pretty good evidence that Bordetella pertussis can form biofilms in infants or in humans. So what we did was we collaborated with our colleague here at Nationwide Children's Hospital. And what they do is they actually take lungs from patients who die of normal patients, and then they freeze these lungs, and then they take sections of these lungs and they grow in these filters where the cells are growing on these filters, and you have the apical surface and the basolateral medium. After about 6 weeks of growth, you can see these cells are actually not only very well differentiated, they form columnar cells, they're basal cells, and then they actually form the cilia. These are actually ciliated epithelial cells. So if you look at by scanning electron microscopy, uninfected culture, what you see that these cells are actually forming cilia, and you can even detect the beating cilia. The cilia is actually beating, and you can measure the ciliary beat frequency. So when we infected these cells with Bordetella pertussis, what we saw was that Bordetella pertussis after 24 hours can form these biofilms or aggregates or tangles of bacteria. And again, I want to emphasize that these bacteria are attached to the cilia, which is the characteristic of infection of Bordetella pertussis. We also use confocal microscopy, where the bacteria are stained green, the BPS polysaccharide is stained red, and eDNA is stained blue. I don't have to tell you these masses of bacteria that are forming with very striking structures. At some places, we see bacteria co-localizing with the DNA and the BPS polysaccharide. At some places, the DNA is sticking out. And at some places, only the BPS and eDNA are actually forming complex, which is not surprising because we believe BPS might be positively charged and eDNA, as you know, is negatively charged. So what we did was we also compared the delta BPS, how it forms a biofilm on these cells, the delta BPS strain. And as we found from in vitro abiotic surfaces and mouse, that compared to the wild type strain, the delta BPS strain was essentially not forming any biofilms. So what we have done so far, I've told you, is that we have developed mouse models of biofilm formation and a human relevant models of biofilm formation. And we are currently studying using these models to study better the determinants and the requirements for Bordetella pertussis biofilms. So I want to tell you a little bit more about the role of Bordetella pertussis in pathogenesis. So what we did was we infected the mice with wild type and delta BPS strain, and we measured the CFUs. We enumerated the CFUs from the nasal septum and the lungs. And to our surprise, strikingly, as early as 1 day post-infection, and I think we published even 6-hour post-inoculation, there was a defect in the colonization of the delta BPS strain. And by 3 days post-infection, the delta BPS strain was essentially cleared from the nose. And this was striking because this was the first report of a Bordetella pertussis factor that was involved in the colonization of the nose. With the lungs, we saw there was a 1 log fewer bacteria in the lungs, and it took quite a while for the lungs before it got cleared of bacteria. So we believe that BPS is required for colonization, exclusively colonization of the nose, but it also promotes colonization of the lungs. So we asked if BPS is functioning as a specific nasal adhesin. That means, is it promoting attachment to the nasal nasal cell lines? So what we did was we took cell lines A549 is a human epithelial cell line from lungs, and RPMI 2650 is a human cell line from a nasal carcinoma, patient with nasal carcinoma. And what we saw, which is consistent with our colonization data, that the Delta BPS strain was did not really promote colonization of or attachment to the lung cell line, but it was required for attachment to the nasal epithelial cell line, suggesting that BPS is a specific nasal adhesin. So this is, I want to show you some exciting data, and this is very new data. We are writing up the manuscript and hope to submit it soon. What we did was we took the BPS locus and we cloned the BPS locus in an E. coli K12 strain, and the E. coli K12 strain now produces BPS. And this E. coli K12 strain is your typical DH5 alpha or MC265, which is a non-pathogenic strain. And what we saw, since E. coli produces the PGA polysaccharide, which is similar to BPS, we took a strain which was deleted of the E. coli polysaccharide. And we either clone the vector only in the strain, or we clone the BPS containing plasmid, locus containing plasmid. And what we saw, if you infect mice with either the vector and the BPS containing plasmid, the vector containing strain did not colonize the nasal septum at all, but when you have the BPS production, it colonized the nasal septum. With intranasal infection, we saw it after 4 days of inoculation, we saw it colonize very efficiently both the nasal septum and lungs, which is suggesting that you can express a single virulence factor in E. coli and confer the ability to colonize or enhance survival in the mouse and lungs. And we believe this is pretty striking data because there are very few reports of expressing a single virulence factor and confer the ability of pathogenesis or virulence in an animal host. So finally, I want to address another question is, why is whooping cough coming back? So one of the few findings that have come out in the last few decades is that the circulating Bordetella pertussis strains are actually showing antigenic and phenotypic drift. That is, compared to the laboratory adopted strain, some of these circulating strains are losing vaccine antigens, or they have polymorphisms, or their phenotype is changing. So our hypothesis was that pertussis resurgence is linked to variations in the biofilm capacity of circulating Bordetella pertussis strain. So we got hold of some strains of Bordetella pertussis isolated from 2008 to 2014. And in this, the suffix actually indicates the year that the strain was isolated. And when we grew them in the roller drum, we saw that compared to the wild type laboratory adopted strain, all these strains formed a very thick and robust ring at the air-liquid interface. We measured the bacteria present in these rings, and we found that all these circulating strains were actually forming biofilms. So we think that the reasons these strains are circulating and surviving is because of the biofilm enhanced biofilm capacities of these strains. We tested the adherence of these circulating strains and to A549, and we found that some of these strains actually attach better to the lung epithelial cell line. And also when we measured the attachment to the ciliated NHBE, the human bronchial epithelial cells, we saw that these strains are attaching better, suggesting that they are adapting or evolving to increase the attachment. And we are hoping that these strains would display enhanced colonization in a mouse model. So I will end my talk with that by acknowledging the people. You cannot do any science without graduate students, postdocs, and I've been fortunate to have some really brilliant students in the lab. And most of the work that I presented currently is being done by Audra Fullen and Jessica Fairman. Han is a new graduate student in the lab. Galen is an undergraduate, and Amo is a postdoc who just joined the lab a week or two ago. We collaborated for the NHBE cells with Mark Peoples at Nationwide, and his technician Phil Chen helped it. And also for staining and characterizing the NHBE cells, we collaborated with our colleague Estelle Cormet Boyaca, and with Dr. Purnima Dubey, we have an ongoing collaboration with immunology and vaccine studies of Bordetella pertussis. And I've been fortunate so far to be funded from OHU, NIH, and OSU. With that, I will be happy to take questions, and I would stop sharing my screen.
Thank you so much, Dr. Deora. It was such an interesting talk, because I don't know, like, when I was listening, I came two questions in my mind previously. One is, why is there some data in India? What is the ratio? Like, is there pertussis death still exist? And another is, that's part of polysaccharide, because you were saying that beta 1,6 glycosidic bond exists there between the N-acetyl glucosamine. So is it like uronic acid substituted, or some, like, is there in substituted functional groups? Because often the pathogenicity also increase for the substituted functional groups. So is there some reports on that available on BPS polysaccharides?
Yeah, so those are good questions. I don't know the latest data with India, but when I have visited India, when I talk to the colleagues there, they actually say in India, they, like everywhere else, they actually don't diagnose for pertussis. So almost every country, the cases reported are actually tip of the iceberg. So I'm suspecting in India, there are also cases, but they are just don't go reported. And also in terms of the polysaccharide, whether it's substituted or not, so far there's no report of the PNAG polysaccharide family that there is any substitution. There was a report of there was some succinyl groups with the polysaccharides, but I think that was proven to be incorrect. But what interesting modification of this polysaccharide is it has got a deacetylase gene. In our case, it's a BPSB. So what happens is this polysaccharide, after it's expressed on the surface or displayed on the surface, the acetyl groups are removed by this enzyme, which is part of the locus. And that has implications for pathogenesis, which although we don't know for our polysaccharide, but for other polysaccharide, it has been shown that the groups are actually positively charged. So when you think about a positively charged molecule on the surface, let's say, for an example, antimicrobial peptides. Many antimicrobial peptides are positively charged. These are innate immune components. So one of the hypotheses is maybe the reason it's resisting antimicrobial peptides is because it's repelling. So also it can function as a shield to shield them from the killing by innate immune components. So yeah, we don't know. We are currently in the process of determining the structure of our polysaccharide. I mean, I suspect it's going to be the same as other polysaccharides, but if there are any modification, that would be really exciting.
Great. Actually, I didn't have even idea that in Chile, pertussis deaths are, like, pertussis cases are so much. This is very interesting because there is vaccine and still the cases are increasing. So it's really something. And probably when it starts, as you were saying, that the graph gets increased, and hopefully it will not be something like that.
Well, I hope so, because I didn't know till I started looking for it that they switched to the acellular pertussis vaccine in 2018. And 2020, there's an increase. So I hope it's just a blip, but we will see in the next decade or so.
Dr. Deora, there are some other questions in the chat box too. One from Dr. Anshu Bharadwaj. BPS is unique to Bordetella pertussis, and two, is circulating strains evolved from environmental strains?
What was the first one? BPS is what?
Unique to Bordetella pertussis.
So, like I said, BPS appears to be the part of the family of polybeta 1,6 N-acetyl glucosamine polysaccharide. This is expressed by a number of bacteria: Staph aureus, Staph epidermidis, E. coli, Acinetobacter baumannii, Bordetella pertussis, Bordetella bronchoseptica. So the family of polysaccharide is not unique to Bordetella pertussis, but like I said, the polysaccharide might have different structure. And as far as the environmental, evolving from environmental strains, Bordetella pertussis, unlike other bacteria like Pseudomonas, does not have an environmental reservoir. So when you are thinking about clinical strains evolving, at least for Bordetella pertussis, it is exclusively for the evolution happens inside the host. And one of the things we have found that the circulating almost about more than 1,000 pertussis strains have been sequenced. Every single one of them has the intact polysaccharide locus. And what we have found that most of the strains are, all of the strains we have tested are actually producing the polysaccharide, and they actually are producing more polysaccharide, higher amounts of polysaccharide than our laboratory adopted strain. So that's what we are focused on right now, studying the clinical strains.
Okay. There is another question by Srobhana Sarkar. Other than biofilm, what are the factors why acellular pertussis vaccine does not work?
So certainly the factor is what they are finding, that there's a pressure from the vaccine. The strains are evolving. Like I said, what they found that many of the strains that when they sequence it and when they characterize it, they are not expressing, they are completely missing the production of one of the vaccine component called protactin. Also, many of these strains are producing higher amounts of the vaccine components like pertussis toxin, FHA, protactin. So the vaccine is becoming ineffective. In terms of immunology, one of the very interesting things they found that for efficient protection from pertussis, either in mouse or in humans, you require a Th1 type immunity. So Th1 type immunity is required for efficient protection. So that wholesale pertussis vaccine that we were using, that probably most of the older folks like me got the WPV, was actually generating a Th1 type immunity, and it was effective. It was very effective vaccine. But what they found that the acellular pertussis vaccine contains alum as an adjuvant. And alum is known to generate a more biased Th2 type immunity. And at least for pertussis, generation of Th2 immunity is not that effective. So one of the projects in the lab in collaboration with Purnima is we have discovered a protein adjuvant, which actually switches the immune response of the acellular pertussis vaccine in the laboratory to more of a Th1 type. So currently we are studying that adjuvant along with identifying other vaccine components.
Okay, there is another question. I will make the translation for you for that. Daniel Valenzuela. It is like, has the colonization been studied in the presence of bacteria typical of the aerial epithelium? And the second is, how is the interspecific competition capacity of Bordetella pertussis?
So when you say colonization has been studied, so it has been studied. Yeah, so in the so for in the human bronchial epithelium that I showed, we are right now studying different mutants and trying to determine whether they are involved in attachment and biofilm formation. There have been reports people have used these epithelium. Attachment colonization has been studied in cell lines, like I've shown you with A549 and RPMI. Also, it has been studied in mouse models. Mouse models is extensively used in pertussis research. Recently, they have developed a baboon model for pertussis, non-human primate. And one of the really nice and unique things about baboons is that when you infect baboons with pertussis, they actually cough, which is one of the symptoms that is not seen in mice. So the baboon model has been developed by one of our colleagues at the FDA, and they are studying it not only for understanding pathogenesis, but also for vaccine efficacy.
Okay.
And what was the second question?
The second one is, how is the interspecific competition capacity of Bordetella pertussis?
What do you mean by interspecific? Are you talking about species-specific competition?
I think that I think that I'm not even sure if it's intra or inter, but it is written interspecific competence among different species probably.
I don't understand the question. I mean, there have been 1 or 2 reports of where they have actually treated mice with antibiotics and got rid of the microbiota, and they have seen higher colonization. But other than that, I'm sorry, I don't understand the question. And if she or he can contact me later, and I should be able to answer it.
Okay. I think there are no more questions in the chat box. So if there is no more questions, we thank Dr. Deora for his important time and accepting the invitation to talk in this event. We are so much thankful to you, Dr. Deora. And please be online for the next talks if you have to listen. And also the link is given there for the YouTube link that is going on live. So we will go for the next presentation now.
Well, thank you.
Thank you very much, Dr. Deora, for your excellent presentation. The second presentation of today, on this morning, will be in charge of the principal organizer of this webinar, Dr. Aparna Banerjee. First of all, we want to say thanks for organizing this event, and it's a great work what you do. Dr. Banerjee is a PhD botany from the University of Baldwin, India. She has bachelor's and master's in biotechnology. She has done her postdoctorate from Universidad Católica del Maule, Chile, where she is presently working as assistant professor. In her 7 years of research experience, she worked on extremophilic microbiology and microbial bioactive compounds, specifically polysaccharides. Her research interest also includes biodiversity, conservation, and bioinformatics. She is presently the Joint Secretary of India's Bioinformatics Society, BioQuest. She is a principal investigator of the Fundeseed Initiation Project that deals with bacterial polysaccharides from Chilean hot springs and their application as food additives. She is also co-investigator in 2 other projects in Chile. She has more than 25 publications in nationally and internationally reputed journals and has been awarded of the Indian Science Congress Association Young Scientist Award. Welcome, Dr. Banerjee.
Thank you so much, Alex, for your nice introduction. It's always nice to listen about ourselves. So basically, thank you, because I have not been the principal organizer of the event. We both have organized the event, and hopefully we will have a nice time. So I'm going in the presentation mode. I hope it is full screen now. Okay.
Yes, it is.
Okay, thank you. So today we are going to, in the international webinar in the story of microorganism in the context of International Microorganism Day, I'm going to talk on exopolysaccharides from hot spring bacteria as food additives. I will discuss about both the works that I have done in India and also now I'm doing in Chile. So I will just give a glimpse of that. Well, so I'm presently working as assistant professor in CIEAM, Universidad Católica del Maule, Talca, Chile. As we start, let me tell that microbes are omnipresent. In the last talk, we were listening to Dr. Deora, and I was so much interestingly listening to him that polysaccharides produced by pathogenic bacteria, they are forming a biofilm network, and that is ultimately related with pathogenicity. It's a factor of survival for the microorganism also against different external compounds that can affect the growth of the microorganism. But indeed, microorganisms present in different others environment also produce polysaccharides. So I'm going to talk about polysaccharide produced by the microorganism that is surviving in the extreme environment, that is not associated with pathogenicity, but it is solely associated with the survival of the microorganism. And also they are industrially important. So I will focus one of their industrial importance, that is in food industry, as food additives. Well, so microbes are omnipresent, and where not? Like when we talk about microorganism, probably the things that comes to our mind is microbes are present in air, they are present in water, something like this. But let me tell you that microbes are present in all the otherwise inhospitable environment where we think that the life form cannot be present, like permafrost region, polar region, cold mud volcanoes, hydrothermal vents, hot spring, fumarolas, hyperacidic lakes, desert environments, that is arid environments, even nuclear contaminated sites, deep sea hydrothermal vents, and where not. So all these otherwise inhospitable environment where the life forms are not present, microbes are not only present and growing, they are thriving over there. And actually, when we see this kind of extreme environment, if we go near that, we can see colorful biofilm formation over there of different microbial colonies. So today we are going to focus on hot springs, one of the extreme environment in both India and Chile, and the microorganism thriving in hot spring, producing polysaccharides as a component of their biofilm and its relation to food industry. Well, so when we talk about microorganisms that are in extreme environment, they are surviving, they are extremophiles. That is the basic idea that comes to our head. Microbes surviving in the extreme environment are extremophiles. So if we talk about hot spring, there are several factors. One is the nutrient concentration is always very low, low oxygen availability, the temperature is extreme, and the added factor is species diversity is most of the time relatively unknown. Then comes the concept of polyextremophiles. What is polyextremophiles? If the extreme environment have more than one extreme factor, for example, if we talk about Chile, it is located in the southern hemisphere region. So we know that solar ultraviolet ray is heavily present in the southern pole. Extreme pH can be, a lot of heavy metals can be present, and extreme temperature. So a lot of factors are basically in inducing the growth of the microbial biofilm in this kind of environment. And because the extreme factor is more than one, so they are called polyextremophiles. If I give the example of India, the hot spring that we have worked, Panifala hot spring in West Bengal. So there was extreme temperature, there was extreme heavy metal. I will again talk about this. So again, that there is more than one extreme factor. So we are calling it polyextremophile. That is the little basic difference between extremophiles and polyextremophiles. So basically all these extremophiles can be acidophile, thermophile, halophile, cyclophile, bacteria surviving in arid environment, in UV contaminated region, or alkaliphiles. They all have their own cellular setup to fight against the extreme environment. For example, in case of alkalifile or acidophile, they have a lot of proton pumps. They have a lot of antiporters present in their cell that they can take out and take in the ions inside or outside of the cells. In case of halophiles, they again have the antiporter present. They have proline betaine, which acts as an osmoprotector for accumulation of the ions. Then in case of thermophiles, we know for the stability of the cell membrane, they have some isobranched chain fatty acids. In case of cyclophiles, again, the cell membrane structure is different. In case of UV resistance, they have added DNA repair proteins. So in case of all the different extreme environment, the cell have different set of protection strategy. One protection strategy that is very common to all of them is basically the biofilm formation. Well, so when we thought about the tree of life, it's basically bacteria, eukarya, and archaea. And out of these three big, the three domains of life, bacteria, eukarya, and archaea, when we look at eukarya, so basically eukarya, they have very less diversity in terms of growing in extreme environment. Basically, some algae, they are halophiles, reported to be halophiles. Some fungi that are reported to be psychrophiles and thermophiles. When we look at the archaea, we have the idea of archaea. If we talk about the idea of Yellowstone National Park in United States. So basically in Yellowstone National Park, that is a model for studying hot springs. And archaea, we have the idea that they basically grow in a very high extreme temperature. So they can be piezophile, hyperthermophile, a little of radiation resistant. But if we look at the bacteria, they are really, really very diverse. They have all hyperthermophile, thermophile, psychrophile, piezophile, alkaliophile, acidophile, halophiles, radiation resistant. So bacteria are really much diverse and thriving in all the possible extreme environment around the world. I will just give a little introduction that throughout the different time period or decades, there have been excellent works that have changed the pathway of how we talk or think about extremophilic microbiology. The first work was there in 1972, like the first elementary work on Yellowstone National Park, thermophiles, was reported the bacterial diversity in bacterial stromatolites of the hot spring and geyser of Yellowstone National Park. Then there was a report, if we talk about Chile, probably for all the international participants over here who are present, the first place that comes to our mind is Atacama Desert. And Atacama Desert is really the dry limit of microbial life. It is the dry limit of life actually. The driest point of the earth inhabits in Atacama Desert. And the soil has been studied a lot for Mars-like soil. And the report was in 2003, one of the very elementary report of how Atacama Desert shows dry limit of life and mimic much like the environment of Martian soil. When the recent report came in 2020, probably you all have studied that. It's Improbable Oasis, published in Science magazine again, that discussed about a Mexican lake. So in different decades, different extreme environmental system around the world have been reported for its microbial diversity. And this is important because if we don't know the diversity of the life form in the extreme environment, in an era of ever-increasing climate change impact, probably we can lose them, or probably they have some industrial importance. Even before knowing them, we can lose them. Well, so in all this extreme environment, how they adapt to fitness? They can have loss of function mutation. The protein stability is very different in different systems. They produce a lot of different stress response enzymes depending on the stress factors. The membrane fluidity, as I told, are different. And definitely one unique point is they all form biofilm networks. So in environmental pressure, exopolysaccharides or polysaccharides, basically, they are produced by the bacteria. What these exopolysaccharides do, they permit adhesion. And after they are getting adhered to a substrate, they divide and they form biofilm. And this biofilm ultimately gives the microorganism protection against different stress factors like temperature, salinity, UV ray, different toxic compounds, for example, antibiotics for the pathogenic bacteria, osmotic stress, and a lot. So just for giving a small, like how to understand it more simply, when we human feel very cold, what we prefer to stay in the home with the heater on. When it's very hot, we try to be in the home with the AC on. So basically microorganisms in different extreme environment, they have their own protection strategy. So this biofilm basically act like an umbrella that the microorganism can survive and thrive in the extreme environment. Well, so these exopolysaccharides are not only a factor that helps the microorganisms to survive in an extreme environment, but they're really, really having a lot of industrial importance. They have a lot of industrial importance to talk in food industry, in wastewater treatment, in pharmaceutical industry, in biomedicines. But today I'm going to talk in the food industry. And depending on the different industrial applications, their properties are different. So for example, for food industry, they need to be a structural material, a biopolymer or a biomaterial. They need to have an emulsifying capacity, thickening capacity, gellifying capacity, or added with some biological activity. So we are going to discuss all this with our works. Well, and some additional secondary activity that they can also have for food processing is they should be a coating or packaging material. So the first case study from India, during my doctoral study, what we have worked, we worked on a hot spring that is located in West Bengal, India, in Asansol region. So this is basically, it's in the Sonata belt of geothermal region in India. There are 6 geothermal belts. Out of that, Jharkhand, West Bengal geothermal belts comes under Sonata region that has been on Jurassic age for the volcanism that happened in the Jharkhand, West Bengal region. So this Panifala hot spring located near a coal mines region. The temperature was nearly 66 degrees. pH was a little acidic, and the arsenic, very, very interestingly, it was very high, around 433 parts per billion. Earlier, there was arsenic-rich hot spring reported in India, but was from Himalayan Manikaran region, and that was nearly 130 ppb, 133 ppb parts per billion. So it was nearly 3 times more than that. So the reason that can be because this Jharkhand, it's just a theoretical overview that why they are having that much high content of heavy metals, because probably they are related to a Jurassic age volcanic activity. So the ground water connection of the hot spring is still now connected with the heavy metals that is present during the volcanism. So probably they are coming from the rudimentary rocks during the volcanism. So, well, the temperature is high and the heavy metal content is high also in this extreme environment. We have actually published a lot of other works other than food industry. I've just put it here that I'm not going to talk today. I'm only going to focus on one particular application that we have found. So we have basically first reported the exopolysaccharide structure of Bacillus anthracis. This is a highly pathogenic strain, and we need to work on BSL-3 laboratory. But this was also the first report of one Bacillus anthracis that is a non-pathogenic variant of Bacillus anthracis. So we have also published different other works other than these, where we proved by the whole genome analysis and by cellular and molecular analysis in mice model, in cell line, how the Bacillus anthracis is unique and it is non-pathogenic. But well, this Bacillus anthracis is interestingly producing exopolysaccharides. And this exopolysaccharide that we got maximum was nearly 8 grams per liter of dry weight in a nutrient supplemented medium. It was a low molecular weight exopolysaccharide, and they were highly carboxylated also. So when it is highly carboxylated, they have free negative charge and they have more propensity to bind with the ions or to interact with the environment. So they're very, very interactive. Also, because this Bacillus anthracis was reported from a extreme environment or a hot spring, so probably it was also having an added thermostability. The melting transition started at 276 degrees centigrade, which is really very high thermostability. With different chemical analysis of gas chromatography and NMR spectroscopy, we confirmed that only glucose was present at the monosaccharide component in the EPS. And well, from the NMR, it was confirmed that because it was having alpha-1,6 glycosidic bond, so it was definitely a glucan. But as we confirmed, that from the 2 to 5 ppm, the different anomeric proton resonances of dextran, so we concluded that this exopolysaccharide produced by this Bacillus anthracis PFAB2 from this hot spring of India was basically a alpha-1,6 glycosidic bond containing dextran. And dextran type of exopolysaccharide was already reported earlier. Basically, it was reported 200 years back by Louis Pasteur when he saw in a wine bottle precipitate of white precipitate. So he thought that it is like the the teeth dextrine. So it was basically on his ideology, on his respect, the name was given as dextran. And dextran is reported from lactobacillus group. It is already reported from different bacilli group of bacteria, as it is dextran is used a lot in food industry, in biomedicine industry, because it is neutral in pH. So when we see that it is dextran in nature, and we saw that theoretically what applications dextran is having in food industry, we see that this dextran produced by Bacillus anthracis PFAB2 was showing significantly high, around nearly more than threefold of increase in DPPH and ABTS mediated free radical scavenging activity. So it was showing high antioxidant property compared to the standard ascorbic acid. We have also seen that the EPA showed significant amount of emulsification activity against the vegetative oils that are regularly used in food industry. And also we have tried to see its rheological property, or its gelifying or viscosifying property. So what we have seen that at neutral pH, normally in food industry we use neutral pH. So when we have used this pH, neutral pH exopolysaccharide powder in rheology, we have seen the hysteresis area is better. That means the structural reversibility is also better. The phase transition started around 72 degrees. That means that when we increase the temperature and we increase the pressure, the solution of the exopolysaccharide is producing a complex viscosity, liquid-like character, solid-like character, and the elasticity was very interesting. That is very particular for a food industry. So basically, in a conclusion, it was showing thermoresistant shear thinning behavior when we added sodium chloride to the exopolysaccharide. So it was showing, basically, it was demonstrating all the activity that is needed to be used in food industry as food additives. So I will now tell another example of the work that now we are doing. It's a part of the Fondesit initiation project that I am presently working in. Basically, we are working in the Maule region hot spring. As you can see the map of Chile, Maule region is located just in the central Chile, very near to the, it's like 2.5 hours from the capital of Chile, Santiago. And in Chile, the work has been done a lot in the north of Chile and in the south of Chile. The central Chile is very less reported or very less work in terms of the microbial diversity in extreme environments, or the hot springs of Maule region is not yet reported for the part of its microbial bioactive compounds. So we are basically working in some other hot springs. We are working in around 4 hot springs, but some interesting result that we got is from 3 hot springs that I have mentioned. One of this is Campanario that I will talk today. We are basically working on 4 different polysaccharide. I will talk about one interesting on them that is showing the best result among all of them. Well, so Campanario is high in temperature. The temperature is around 56 degrees centigrade. Definitely there is presence of ultraviolet ray, and the heavy metals that are present, because Chile, you all know, Chile is one of the most earthquake prone region and have around 400 active volcanoes around Chile. So all these hot springs are basically formed during the volcanic activity of Laguna del Maule, happened years ago, millions of years ago, and are all volcanic origin hot springs in nature. So they probably have a lot of heavy metals and trace metals present in the environment. So when we have I'm not going to tell which microorganism it is because the manuscript is still under preparation. So we have produced the exopolysaccharide, and we have tried to do the response surface methodology analysis of the exopolysaccharide produced by this bacilli. This is also a bacilli species. So you can see the normal plot of residual and the predicted versus actual plot of residual. And you can see that our factors were 4: carbon source, nitrogen source, pH, and the inoculum size. And you can see the plots varying these 4 factors. And this is the 3-dimensional study, 3-dimensional plot of the RSM optimization. As you can see that the predict test was nearly 6.05 gram per liter, and the actual production was 5.95. So the desirability score was very near to 1. This is how the surface morphology looks. Surface morphology is basically very interesting to have an idea about the structural biomaterial property of a polysaccharide. When the surface of the biomaterial is smooth and very compact in nature, they basically show a pseudoplasticity. That is, the biomaterial can absorb a lot of water, and when it can absorb a lot of water, they can be readily used as a biomaterial in different industry. You can see the same in the atomic force microscopy also, that the surface is very compact in nature. From the elemental analysis and the 3D mapping, it was seen that the polysaccharide was also supplemented with phosphate and sulfate group. When the polysaccharide is basically supplemented with it's a phosphated or sulfated type of polysaccharides because they are negatively in charge, these groups. So basically these groups added antioxidant property to the polysaccharides. There are a lot of reports that normally the marine bacterial polysaccharides are highly phosphated in nature, and they are used a lot in medical as antioxidant agent in biomedicine, in pharmaceutical industry, as food additives. So for the structure and analysis part, we have done its HPLC analysis, and we have seen that the ratio of mannose is to galactose is to glucose was 10:3:1. So basically this was a more mannose-ylated exopolysaccharide reported from this bacteria. And the temperature stability of the exopolysaccharide produced by this bacilli microorganism have started from 192 degree and show a phase transition at 315 degrees centigrade around. So also it showed a high thermostability. As you can see here from the NMR also. So overall, this exopolysaccharide produced by this bacilli group was a mannose-ylated exopolysaccharide, thermostable also. Here you can see the rheological property. We have basically changed the pH from 3 to 5 to 7 to 9. We have done a different concentration of the exopolysaccharide from 1 to 5%. And we have seen that the 2% polysaccharide was showing best rheological property, that is viscosifying activity or emulsification activity. So basically at pH 7, as you can see here, at pH 7, 2% exopolysaccharide solution was showing the best rheological property. So that also detects that in neutral pH, the exopolysaccharide solution was showing viscoelastic property. That is already confirmed from the surface morphology of the microscopic feature of the polysaccharide too. So it was basically confirmed that the exopolysaccharide is a good biomaterial, having a good thickening, viscosifying or jellifying activity. So we have tried to see its emulsification property with compared to synthetic surfactant and also very known EPS biopolymer. Xanthan gum, you all know probably, this commercial exopolysaccharide biopolymer also produced by Xanthomonas campestris. So when our study EPS have basically shown very good result in case of coconut oil, in case of corn oil, in case of canola oil, avocado oil, sunflower oil, and even sesame oil. So a little. So other than olive and sesame oil, basically against a set of vegetative oils that are regularly used in food industry, it was showing good activity compared to the traditional biopolymer xanthan gum. We have also tried to see its antioxidant property in 5 different methods, but 3 was really showing very good result that I have shown here. DPPA scavenging activity, H2O2 and ABDS mediated scavenging activity, which was showing, it is not showing as equal as good antioxidant property like standard ascorbic acid, but was very near to the standard ascorbic acid that is regularly used in food industry. So basically, overall, the exopolysaccharide produced by this bacilli species from a Chilean hot spring was also showing different property as a biomaterial to be used in food industry as an antioxidant, emulsificant, jellifying or viscosifying agent to be used in food industry. So to end the talk of today, I will tell that 17th September was International Microorganism Day, and probably we all know what is the goal of International Microorganism Day. In International Microorganism Day, we are celebrating the microorganism life, the microbial life at the edge of site, their different role in human, in culture, in the environment, how they interact with each other, and how they are impacting our daily lives in the planet Earth. So how about we talk about protecting microorganism? When we see the tree of life, the traditional microorganism is totally focused on plant and animals. Very less talked, or it's like relatively not at all talked to conserve the microorganism, bacteria or archaea. They are not till now added to the traditional conservation strategies. So if we see the publications related to microbial conservation, the first that the in situ conservation that came into limelight was in 1964. Then in 2019, the first report was there about the importance of host associated microorganism and to include them in the traditional conservation goal as an integrative management strategy. So what is integrated management strategy? That when we talk about one microbiome, when we talk about a host, to conserve a host, it is necessary to conserve that microorganism also, because microorganisms are interrelated in a symbiotic way with that host in different factors of the growth of that host. But what about all the microorganisms that are taking part in the biogeochemical cycle, that are present in the otherwise inhospitable environment of planet Earth, or in every part of the planet Earth, playing an important role in its own ecological niche? So we need to talk about microbiome conservation now in an emergent era of climate change that we have published one perspective later on that. So we have to talk more about conservation of microorganisms, not only to conserve them and not only in a time of climate change, but also it is important to know the identity of the microorganisms before we lose them. And also the microorganisms are a hub of different microbial bioactive compounds that are regularly we are using in different industry. For example, if you see a pack of yogurt, if you see a packet of biscuit, if you just move and see the ingredients, you will probably see dextran, you will probably see xanthan, you will see something like this, one polysaccharide biopolymers that is used. So basically they are already used in the food industry, and without even realizing, we are taking them, and we already know that they have the probiotic and prebiotic effect in our health, that is they modulate, it has an immune modulant activity. So it is very important that we conserve them before we lose them. Well, so these are the different images of Universidad Católica del Maule where I'm working. It is really very, very beautiful in different seasons. And as I like to take photo, I try to take photo in different seasons. So, well, I'm thankful to all. I'm really thankful for the part of my doctoral program to Professor Rajiv Bandyopadhyay from University of Bardhaman. The Panifala work that I have shown. I'm thankful to Universidad Católica del Maule, CIEAM, where I'm working now, the laboratory that I'm working, CENBio, and finally the funding, because without funding, science is very boring. So I'm really thankful for Fondesit Initiation Grant to work in Chile. And basically, if I miss anyone, I really don't want. All the people that are associated with my project, I'm really, really heartfully thankful to all of you. The collaborators are Dr. Gustavo Cabrera Barjas from UDT Concepcion, Universidad de Concepcion, Professor Bandyopadhyay from University of Bardhaman, India. And I'm really blessed with amazing students. They are undergraduate thesis students, and they have already published a very interesting article on exopolysaccharide producing glycosyl transferase few times back. It's an in silico work though, but they are very, very curious students. I'm also very much thankful to the project assistant Alaidis Gomez, who is working with me, and all others, Professor Rajesh Sani from USA, Sura Break from Iraq, Dr. Sugunakar Bhuri, Dr. Salini Gordodra from India, MS Trobona Sarkar from India, Dr. Alex from UCM Chile, he is also my colleague, and another student, Catalina Marin Sanhueza, she's also working on the biofilm production of this interesting microorganism from the hot springs. So probably someday we can talk about that also. And I'm very, very thankful to you all, and I am open to questions if there are any. Thank you so much.
Thank you very much, Dr. Banerjee, for your great presentation. We have some questions on YouTube. The first one is from Trobona Sarkar that says, other than natural extreme environment there are man-made extreme conditions to like different kind of pollution. What might be the possible survival strategy of the microorganism live there?
Okay. Yes, that's a factor probably I missed to talk. Yes. man-made extreme environment is probably also the antibiotic resistance that is growing every day, or probably man-made extreme environment is acid mine drainage that when I was showing the image of the microbes are omnipresent, there was given there, or probably any industrial effluent where the microorganisms are living, but it is so toxic in nature that other life form cannot exist there. So even in this kind of condition, when we see from near, many times because of the low oxygen availability, they form a flock kind of place and different colors of biofilm. So even in acid mine drainage or man-made industrial effluents or other extreme environment where there are high level of toxicity, less oxygen concentration, or high level of toxic compounds. For example, it can be in case of industry, it can be heavy metals. In case of textile industry, it can be dye. So these different extreme factors probably induce them to, first, they can be bioaccumulated inside the microorganism. So definitely they have a lot of antiporter pumps. And the second is probably they can be producing biofilm to survive under that extreme environment. Yes.
Thanks. A second question is from Dr. Anshu Bardwaj. I'm sorry for the pronunciation. And he asks, how do you take the innovations in food material downstream? What kind of regulation apply?
Okay, for the downstream processing. Look, personally, if I say I have really not went to the downstream processing of the polysaccharides yet, probably it need different scale-up processes. We are still on the level 1, so we are not yet done with that. But probably my plan is the next that I'm going to do is scaling up it with fermenters. And because now with the response surface methodology, that we know how much it is producing, we know how to supplement the media. So probably now we are planning to go into producing the polysaccharides in a fermenter, and definitely during scaling up the production percentage will also change. And then comes another factor to try the cytotoxicity. So these are 2 factors that are in my mind that I will do in future, but really till now I have not done that. No.
Our last question is made by Dr. Devashish Mandal, and he asked, have you find any structural difference in case of bacterial polysaccharide grow under different environments?
Yes. I have not talked about the work that I have also done in the samples of Southern Ocean Antarctica that my PhD supervisor have participated in 2011. So we have also published a couple of papers on the Antarctic microorganisms. Well, in both the cases, the factor is extreme temperature. But in both the cases, we have seen that in case of the Antarctic microorganism, that is the marine microorganism, it was more phosphated in nature, more sulfated in nature compared to the, so the compounds that are like the supplemented groups in the polysaccharides, they have more active groups present. The sulfate component in the marine exopolysaccharide was really high. And the second thing that I realized is probably that can be depending on the environment, because the hot spring that we have worked in India, the Panifala hot spring, that can be even a man-made factor, because we all know even in India or in Southeast Asia, hot springs are really treated like a tourist spot. People go to take shower there, and we are destroying the environment. So the biofilm that are existing over there for million years, we are destroying them. So the structure of the polysaccharide produced by these bacteria are relatively simpler. But when we have seen the microorganisms that are present in the extreme environment in Chile, for example, that I am now working, the structure of the exopolysaccharide is much more complicated. They are not simple dextran or glucan or levan kind of polysaccharide. They have different substitution ratio among different group of monomeric polysaccharides, monosaccharides. So they are relatively more complicated. So one thing can be environment, and another thing can be the presence of the microbial biofilm too. So yes, they are different in different environments.
Okay, Dr. Banerjee, thank you very much for your extremely interesting presentation.
Thank you so much.
And now you again, please introduce the next lecture.
Okay, so we will go to the third talk of today's webinar. The third talk is by Dr. Anshu Bharadwaj. She is a biologist and data scientist. She has obtained her PhD in 2008 in understanding genotype-phenotype conditions in human mitochondrial diseases from CSIR-CCMB, Hyderabad, India. She has over 40 publications to her credit and has trained over more than 500 young students. She served as an associate scientific advisor to Science Translational Medicine and is on the editorial board of several internationally reputed journals. She was one of the 4 young innovators in India, UNDP, selected for the prestigious International Visitor Leadership Program by US State Department, awarded Newton Bhabha Fund from the British Council and the Royal Society of Chemistry, UK, a long-term group leader fellowship by CRI Paris. More recently, her lab has developed a unique scientific video game for awareness of antimicrobial resistance, which is endorsed by the WHO India too. Her passion is towards biomedical big data analysis platforms with focus on understanding infection and rare disease biology for better therapeutics and diagnostics. So, Dr. Anshu Bharadwaj, please.
Yeah, thanks, Dr. Aparna. Am I audible?
Yes.
All right. And okay, let me just start by sharing my screen. Is my screen visible?
Yes, sir. Yes.
All right. So first of all, thanks a lot for having me today to share the work that we do in our lab on the International Microorganisms Day. It's been a real privilege. And thanks for the nice introduction, Dr. Banerjee. It's really cool. So what we shall do today is, as you can make out from the title, we will be discussing how do we target biofilms to address antimicrobial resistance. Now, the previous 2 speakers have already given us a very good overview of how biofilms are important from a vaccine perspective or from the perspective of food materials, or basically utilizing microbes and or understanding microbes in different missions. What we will do today is to understand that how one can develop peptides that can actually target biofilms and can allow us to proceed forward in eliminating some of the very hard to treat infections. As Dr. Banerjee mentioned, my lab has developed a scientific video game on antimicrobial resistance, and towards the end of my talk, I'll briefly tell you what it is. So tune in, just stay tuned for that part of the presentation. I'm pretty sure the students in the audience would really like it. So going forward, we all know that antimicrobial resistance is one of the top 10 global public health threats facing humanity today. And it's not discussed as much as the COVID-19 pandemic, and that's why it's referred to as a silent pandemic. The reason why we say it's a silent pandemic is because of the kind of impact it has on the healthcare system per se globally. If we look at the timeline of when the antibiotics were discovered over past 60 to 70 years, if you can see from the top panel and the bottom panel, when the identification of antibiotic resistance happens for these antibiotics, you can actually make a clear observation that discovery of antibiotics is rapidly followed by emergence of resistance against them, which means that we will have to keep coming up with new mechanisms to develop better antimicrobial agents and be more judicious in using what we have today, right? The reason being that if we don't learn to do that now, this is what is anticipated. AMR is a grave threat to human and animal health. These are estimates that are published a couple of years ago, which says that if we are not taking hard steps right now, we'll be dealing with a situation which is going to be extremely difficult to tackle at that point in time. So before we go any further into how do we understand biofilms, let us start understanding what antimicrobial resistance is. So to simply make you understand what AMR is, you have a microorganism, a bacteria. When there is no antibiotic, it proliferates. So obviously it multiplies. But in the presence of an antibiotic, either it leads to the death of the bacteria or it actually slows down the growth of the organism, which either means that antibiotic is not letting bacteria to multiply. On the other hand, there are resistant microbes or resistant bacteria who, even in the presence of antibiotics, can very happily multiply. How do they do that? So there are several mechanisms which, believe me, I've been reading them for many, many years. It's just too complex. They are pretty intricate and they vary from organism to organism. But what do? They try and block the entry of the antibiotic. They try and pump the antibiotics out of the cell, or they destroy them. Other mechanisms include modifying their targets. So say, for example, if you make a change in this protein structure, this antibiotic is no longer binding to the protein target it was supposed to bind to. And likewise, there are n number of mechanisms involved, and it's a pretty intricate process. Our understanding of AMR is still it's still very, very basic. Reason being that most of our studies are focused on planktonic state, as in free-living microbes. And despite the fact that 99% of microbial life is not planktonic. It is in the form of biofilms, and I think the previous two speakers have already highlighted that. So in a planktonic state, our single cell as free-floating entities, and they are mostly responsible for acute infection in humans. On the contrary, biofilms are sessile communities, as you already have seen in the previous presentations. So they come and adhere to a substratum and generate a matrix, which is called as an extracellular polymeric substance. Now they are extremely adapted to a very rough environment. So they actually show extreme phenotypic adaptation. They are very, very old. They are mostly polymicrobial in nature, which means that more than one microbes actually come and stay together in that biofilm, so that they have an option to share genetic material, and together they can be much more powerful. And they are responsible for mostly chronic and persistent infections. The take-home message is that residents of a biofilm can be up to 1,000 times more resistant to conventional antibiotics than their planktonic forms, which means that we need to understand the biofilms in a much better fashion if we are to deal with difficult to treat infections. And as I told you before, it's an ancient phenomenon. Primarily today is being heavily driven by injudicious consumption of antibiotics. And what you see here is not just a specific type of bacteria showing this kind of phenomenon, but you see it across the spectrum, whether it is Enterococcus, Staphylococcus, MTB, Salmonella, or Klebsiella, Shigella. So you see this phenomenon coming from across the spectrum of clinically relevant pathogens. And the issue is that if antibiotics don't work on these drug-resistant pathogens, it is not just infections we are talking about. When there is treatment of diabetes or chemotherapy or organ transplantation, dialysis, surgical procedures, all of these are basically all the secondary infections in these cases require antibiotics. So you imagine a situation when these antibiotics are not working, all of these healthcare system can fall flat on face because none of this is will be then possible. Now what happens? So there is a concept in biofilm which is called as recalcitrance, which means they are extremely hard to treat. They are adaptive in nature, which means that when the biofilm breaks open, the drug susceptibility of the cells within the biofilm might be restored. It's not always the case, but it is restored. The conventional resistance mechanisms, as we saw a couple of slides earlier, have very limited contribution in biofilms recalcitrance. On the contrary, what happens is that it actually have a very multicellular strategy, which means what? The extracellular matrix delays permeation of antibiotics. There is a presence of persistent subpopulation within the biofilm community, and this is a huge problem when a lot of antibiotics don't work on biofilm. This is one of the primary mechanisms. Then antibiotic degrading enzymes like beta-lactamases, they accumulate in the EPS matrix and render antibiotics inactive. And given that within a biofilm community, the population is heterogeneous, like there are several microbes, and they are at several levels of metabolism and growth, they are not vulnerable to any single antibiotic. So now you imagine you're dealing with something which is like a group of pathogens coming together and also not in the same state. And where do you find them? They are practically ubiquitous. You talk of any medical device, whether implanted or urinary catheters, mechanical ventilators, intravascular devices, biofilms are everywhere. If you talk of infections, cystic fibrosis, dental plaque, chronic otitis media, they are actually omnipresent. They are present in a very large number of hospital devices as well as in human infections. And just to take you a little bit deeper into how a biofilm look like. So basically what happens is there are planktonic bacteria which are free floating. They try and identify a surface. Initially it's a reversible addition, which then turns into an irreversible addition followed by colonization. So microcolonies starts to take shape. And then these microcolonies convert into a full-blown biofilm. And the whole point is these multiple microbial species come together as a single biofilm. It allows them, all these microbes within the same biofilm, to have a much enlarged gene pool. They are more efficient for DNA sharing, which means that drug-resistant mechanism of one bacteria can now be shared with the other one. So together they become much, much more powerful. And that is why planktonic cultures cannot be used as a proxy measure of biofilm growth. And I think the first speaker today made it very, very clear that why do you need more human-like systems for biofilm growth to make an assessment of what they might be doing inside the body. And that is the message here as well. If you look deeper, if you look at, there is a gradient of yellow going from outside to inside, which tells you that these bacterial cells within a microcolony, they are across a gradient. The ones which are on the periphery are metabolically more active. As you go inside deeper into the biofilm, these are more or less persistent populations. And then there are water channels that run across the biofilms for the biofilm organisms to take nutrition on oxygen. Depending on the state of metabolism, like say for example, the metabolically active outer layer is susceptible to fluoroquinolones, whereas the slow growing inner layer, these ones are susceptible to lipopeptides. So the point is one has to deal with several antibiotics at the same time to come up with a solution for antibiofilm treatment. So where do we look for solution? What can destroy a biofilm? So what we thought that we can actually focus on antimicrobial peptides. They have been gaining a lot of importance in past decade in terms of treating drug-resistant bacteria. So we thought, why not try it on biofilms? And the examples we have is like cathelicidins are a family of antimicrobial host defense peptides. They are known to disrupt biofilm structures. So what we decided is, let's look at some of the known antibiofilm peptides, see if we can learn their properties and make a reasonable objective assessment of which peptides can have a biofilm disrupting property. So at the time when we started this work, a database which is called BAMS, Biofilm Active Antimicrobial Peptides, just got published in Time. And this is a database that provides antimicrobial peptides which have been tested in vitro and in vivo in a very large range of more than 50 gram positive and gram negative pathogens. So you know that these antimicrobial peptides have anti-biofilm activity across a wide range of pathogens. What we did after that, which is a little unusual, is we generated data set. Normally in a machine learning kind of a scenario, when you want to develop or you want to train a machine to identify peptides or anything with a specific property, you say, okay, I will take anti-biofilm peptides which are active, and I'll say anti-biofilm peptides which are actually not showing any activity. Instead of doing that, what we did is, if you look at the positive data set, these are basically anti-biofilm peptides which are known to target a wide spectrum of bacteria. The negative data set, on the contrary, were quorum sensing peptides that are known to maintain the biofilm. So they are not the ones which are disrupting the biofilm or not showing activity. They are a completely contrasting set. And as you can see in both the sections, we have mentioned which kind of peptides are removed. So we actually removed peptides which were cyclic in nature, or they were having any kind of secondary structure possibilities, or if they were modified. And in case of negative data set, we eliminated any quorum sensing peptide which actually is reported to have a biofilm inhibitory property. So that what we are dealing with is something which is only quorum sensing biofilm maintaining property or have nothing to do with biofilms. And in the positive data set, all exclusive active peptides. Now with this, as you can see, we started with a very big number, but this data set actually regression was done in a very, very meticulous fashion. But the final numbers of peptides we came up with is very small. So what we have to do here is, instead of following an unsupervised machine learning model, we did a supervised machine learning. What do you mean by that? So if you see here, anti-biofilm, we actually gave the machine learning protocol labeled data, which means what? That the machine knew which of them are anti-biofilm peptides, which of them are non-anti-biofilm peptides, and they know which properties are we calculating. So we actually went ahead with the composition of these peptides, the amino acid composition of these peptides. And once you use anti-biofilm peptides and non-anti-biofilm peptides, give it to a predictive model. If we are getting a very good classification on the training set, then we take new data and validate the model. Is the model only working with what it has seen before, or it can work with something that it has not seen before at all? And what we could see is that our model was pretty good, and that is how we came up with the first ever anti-biofilm prediction, anti-biofilm peptide prediction server. And in this, we generated over 1,000 machine learning models, and we have identified the top 6 because they were really good in predicting the anti-biofilm properties of peptides. And if you can see here, there are 6 models. Some of them were developed in Beka, some of them in SVM. And the reason being that we either went ahead with the complete amino acid composition, or we did a feature selection based on which amino acids might be playing more important role in rendering an anti-biofilm property or not. I'll not describe this whole graph end to end. I would recommend that you go and read the paper, but I'll be very happy to answer any questions later on. All in all, what I want to convey is that when you check for the performance of these, the best models on training data sets, you are getting an accuracy as good as 95%. And performance on independent data set, which is a true assessment of any machine learning model, we could achieve similar accuracies. Now, if you see here, I would like you to focus on this particular SVM and the Beka models, where we looked at the first 5 N-terminal residues of anti-biofilm peptides versus quorum sensing peptides, and you see that the classification works pretty much perfectly. Now, what is that the machine learning model is seeing in those N-terminal peptides? We are very curious to see that. Let's see what happens. So if you look at the top panel, these are NT5, which is the N-terminal 5 amino acids of anti-biofilm peptides. These are C-terminal 5 amino acids, and likewise the same for quorum sensing peptides. Now, if you compare the N-terminal 5 amino acids in anti-biofilm peptides versus the quorum sensing peptides, one can clearly see that in anti-biofilm peptides, it's mostly arginine and lysine. But in case of quorum sensing, it is serine. So it is more neutral kind of amino acid, whereas here you are finding more polar amino acids, more charged amino acids, cationic amino acids in nature, right? So there is definitely a positional preference for specific amino acid residues in ABP versus QSPs, and this is what the machine learning model was able to identify. On the contrary, if you look at the C-terminal, the distinction is not so clear, and that is why we are not getting really good model at the C-terminal end. But if you look at the overall distribution of amino acid residues and physicochemical properties in anti-biofilm peptides versus quorum sensing peptides, you could see that the anti-biofilm peptides are abundant in cationic residues like arginine and lysine, and they are polar in nature. They possess a greater net charge than QSPs, and obviously they are hydrophilic in nature because we computed a score which is called as gravy, and most of the values were in negative, which means that most hydrophilic amino acids are represented more in the anti-biofilm peptides. Now, why does this matter? This matters because, as we already heard since morning, the extracellular polymeric substance, or the matrix of the biofilm, is negatively charged. So if there is an there is an antibiotic or a peptide that is positively charged, there is a possibility that it can easily penetrate. However, what we see in case of tobramycin, which has a positively charged and which is a positively charged antibiotic, via some ionic interaction, we don't yet know what, it actually can penetrate, but not so deep in biofilms. It's used, but it's not very good at it. On the other hand, aminoglycosides, which are negatively charged, they actually penetrate very poorly in biofilms. Ciprofloxacin, which is a neutral antibiotic, diffuses very easily through the biofilm, and it is a very strong candidate for antibiofilm treatment. However, it's not the penetration, but the persister phenotype that renders also ciprofloxacin unusable in a very large conditions. So what we are expecting is the antibiofilm peptides are also following a similar property, where they're hydrophilic in nature, and that is what is explaining that how ABPs navigate through the primary aqueous environment of the EPS matrix, and thus may be very good at destroying these biofilm structures. Now, how do we know based on computational models that what we are doing has any meaning? Like what is the evidence? So what we did, we went ahead and made predictions on FDA approved peptide drugs. These are peptide drugs which are basically FDA approved not for biofilm treatment but for other indications. Likewise, we picked up antimicrobial peptides in clinical development. Now, these are peptides that are more drug-like in nature. So if anything gets prioritized here, the chances of taking them downstream is much, much higher. So of the list of FDA approved and ANPs, FDA approved peptide drugs and ANPs in clinical development, we could get positive predictions for a pretty decent set of 29 FDA approved peptides. One of them is shown here, and 10 antimicrobial peptides. I'm showing 6 here. Now, if you look at the first one, this particular drug, it is actually being predicted as biofilm active by 5 out of 6 models. And the indication against which it is used is the respiratory distress syndrome in premature infants. Now, this is a classical case because this drug actually works as a surfactant. And now we know that why it can also have an antibiofilm property. Likewise, if you look at antimicrobial peptides in clinical development and you look at their indication, it talks about catheter infections, it talks about immunocompromised infection in immunocompromised patients, it talks of middle ear infections. All of these have a very strong indication of presence of a biofilm. And that is why we think that we can repurpose these peptides and test them in an antibiofilm therapy. So, it actually merits further testing because this looks very, very promising at the face of it. Now, what we did, after we did all this exercise, I know there are a lot of groups across the world who are trying to develop antimicrobial peptides, antibiofilm peptides. So we actually converted and converged all our models into a web server, making it easy for anybody to use. So if you have a peptide, you feed in here and it will give you predictions on all the 6 models. And as you can see here, this is the peptide I was talking about. What we have also done is, like say for example, you have a peptide which initially is showing that it's biofilm inactive, right? So what you can also do online is you can actually generate mutant peptides. So the software does that for you. And then it also predicts which of these mutations actually are rendering your peptide biofilm active. So you need not synthesize and test. Instead, you can generate an entire library of mutant peptides, do the test, prioritize, and then synthesize that smaller set. We are not saying that take this prediction or just take them downstream, but this definitely allows one to reduce the search space that is available or that has to be spanned out for identifying something that works in an actual biofilm assay. Now, this actually concludes the first part of my talk. And what we also do, we not just use machine learning models, we also do machine learning for therapeutics. We also use machine learning and other methods for diagnosis of infections using genome sequence data sets. But I'll not be describing this work today, maybe some other time. What I would like to share with you in the remaining time I have is the project that I'm really very aggressively working on right now is to generate awareness and understanding on antimicrobial resistance. Now, in May 2015, during the World Health Assembly, all countries who are part of the WHO, they actually came together and endorsed something which is called as a global action plan to tackle antimicrobial resistance. Like, how do we collectively address this huge issue? And if you look at the 5 verticals that were identified, the first vertical says improve awareness and understanding of AMR. The reason is that if everybody does not understand the problem, nobody is going to address it. Like as Dr. Banerjee was referring to microbial conservation in her presentation, that is something that we need to make people aware about. Now, if you understand something better, you know what you can do about it. So what we decided is we would like to do this through a gaming activity. And the reason of doing it through a gaming activity is it has been shown scientifically that if middle-aged, like middle school going kids are taught something, or if they are engaged in an activity, they have a far-reaching impact on how their parents and how their local communities also get awareness indirectly, because they are very, very vocal, they interact. So we thought, why not take something which is more engaging for a younger generation? And of course, all of us like to play games. I'm pretty sure the kids like to play them even more. So we actually then came up with this idea of running an open antimicrobial resistance awareness drive. It's a platform where we have developed a game as well as a bot so that we can, it's a knowledge repository for education and awareness on AMR. What we did. So the first question is, how would you like to design this game? So we looked at the most downloaded games ever. And what we identified is that the most downloaded games have a few properties, like they are very simple to play. There is no complexity. They have very nice graphics and they're very variable graphic. Like there should be more to the eye, you know, there should be something that is more exciting. The interface should be clear. There should be no confusion. There has to be a very clear set of instructions. But the point is, in most of these downloaded games, the content complexity is very low. Now, the content complexity of AMR is pretty high. If we really want people to understand what it is, then we have to work with something which can deal with high content complexity. And that is why we went with a game genre, which is called as tower defense. I don't know how many of you are aware of it, so I have made a small schematic to tell you what it is. Now imagine, like in any other game, there is an end point. Either you are protecting something or you are achieving for a target. In a tower defense game, as the name suggests, you are defending the target. Now in this case, that if you're defending that the enemies should not reach this cross. Okay. Now the enemies start on a particular track. And before they can reach the cross, you need to kill them. So what you do is you plant defenses, and these are called as towers. And these towers will start firing their armors on these enemies and try to kill them before they reach the target. But if the towers fail to do that, and the enemies still make it to the target, then you lose lives, right? Now, it's a very simple tower defense game genre, and this is what we have migrated into something that can explain AMR. Now, how do you do the migration? The adaptation is very simple. The enemies here are pathogenic bacteria. The towers are antibiotic dispensers. The bullets are actually the antibiotics. The terrain type or where it is happening is a biological tissue. Whether you are dealing with lung infection, you are dealing with skin infection, and the target to defend is a capillary. Reason is you would not like something to enter through your body to blood, because once it becomes a blood infection, it actually infects all of the body parts. So the capillary, the target to defend it should not be able to reach blood. Okay, so this is how we adapted. And the advantage with this kind of a tower defense game genre is we actually can deal with very high level content. We can deal with acquisition of resistance itself at several levels. We can do insufficient damage. We can basically tell about horizontal gene transfer. We can talk about antibiotic pathogen mismatch. We can talk about mutation and selection. We can talk about efflux pump, biofilms. So this particular game genre allows us to basically dig deep into these mechanisms of resistance and simplify them in a gaming scenario. All right. And the learning strategy is very simple. that you need to make sure that overdosing or underdosing of the antibiotics is going to lead to drug-resistant bacteria, and even a mild wound that gets infected can prove fatal. So you need to make sure that, so it's a strategy-based game. Don't over-prescribe, don't under-prescribe, just the right amount, and then stop, okay? So right now, AMR has two worlds. One is a skin infection level, the other is lung infections, and skin world currently has 6 levels. So it's a multi-level game. And that is why we thought that, you know, the engagement or the replay appeal is, if there's a single level game, I've played it once, why will I come back to it? So you need to keep the engagement going. So that's why it's a multiplayer game. And what I would like to do with is that I'm going to play a small teaser that we generated for the game so that you know what the game is about. And here it is. So as you can see from this teaser is that the main message is don't overuse or underuse the antibiotics. And as I told you, there were 6 skin levels. Then we have a lung level and 1 COVID-19 level. And let me briefly tell you what it is. So the lung world is basically to convey the message that many, many times you need several antibiotics in combination, like say, for example, tuberculosis. Now, there are several articles that says that TB is a bigger monster than the ongoing COVID-19 pandemic. We just don't pay enough attention there. We need to sensitize the wider community on the fact that one has to take this data on evidence-based, right? The other COVID-19 level is primarily to convey that antibiotics don't act on viruses, and that is where we are trying to say this because there is an absolutely unnecessary prescription of antibiotics in COVID-19 pandemic, and we all know about that. Now, we had to select a virus, and I thought selecting SARS-CoV-2 was easier because there is and not anyone in the world who doesn't know it's a virus. So that is why we thought we'll introduce lung world and the COVID-19 levels. These 2 levels will only unlock if you have crossed all the 6 earlier levels, otherwise they don't unlock. So there is an incentive for you to play the entire game, learn, and then come here. While we were developing this game, we did several play tests in India and in France with school kids, with bigger kids, with college going to schools going kids. And we could identify how and when we need to improvise. So the game has since been improved like 40 times. So we had simplified, there has to be a replay appeal, why do I come back? Because the gaming will actually make levels difficult as you go forward. So you need to be more strategic, but you also have to be hand-body coordination. Then graphics were a very, very critical thing. So we revised the entire game graphics. Content has to be there, but we didn't want to make the game very boring, like overloading kids to remember a lot of things. So a lot of it is built into the game design. So when you win a level, the owl that you see in the game tells you why you did that. And of course, we are in the process of adding more game levels, and music is critical, and every level has a different music depending on how the level progresses. Now, given that we are generating armor for curiosity and awareness, and it is based on sound scientific principles. Currently, it is being developed as a Jigyasa project, which is a virtual laboratory project. The reason being that more than 100,000 students and 1,000 teachers are added to this online platform annually. So this allows us to do web-based playtests on a much larger scale, and this is something that we will be starting off in a few weeks from now. And in addition, the WHO India networks shall also be part of the wider outreach program. What next in the game? So what we are doing now, we are actually introducing the concept of aware, which means access, watch, and reserve group of antibiotics. So in the initial game levels, the player cannot unlock antibiotics in watch or reserve categories. You need to play several levels to have enough money or enough points to be able to unlock watch and reserve, to convey the message that these are not something that you should be using directly. So which means that if a clinician is prescribing a very high-end antibiotic or a last resort antibiotic, the user should know that it's a last resort. Let me try with something which is a first or second choice of antibiotics and not go to the last resort straight away. All right. And if everybody is sensitized to this concept, is when these things might work. On the contrary, what we also thought is it's important for the children to know about these microbes. Like, say, for example, how does it look like? Is it like size-wise, how big it is? Can it move? Is it motile? At what temperature does it exist? How fast it can reproduce? What are the symptoms if you get infected? What are the usual treatment? How long the treatment takes? So what we did, we picked up top 17 clinically relevant pathogens globally, converted all of that into a structured data, developed an AI-based, it's an artificial intelligence-based conversational bot. So we did a lot of machine learning at the back of it with NLU, and converted it into a bot that can talk to people. And depending on how they respond, take them forward in terms of understanding better, in terms of AMR, in terms of different concepts. And the point is that this bot will now be available from within the game itself. So that is something which is an innovation. I have not come across anything that works like this. So yeah, we should be able to see this very, very soon. And again, coming to the point that it's been endorsed by WHO India, and we have used technologies as far as possible free and open source. And the game is available online. The bot with the game will be available soon as well. And yeah, we have been covered nationally by several newspapers. So AMR is currently supported by CIEAM. Initially, it was developed with support from CRI and CIEAM in Université de Paris in France. And we need beta testers. So if you would like to be a beta tester, please let us know. It's basically the point. Give us your feedback on the game. If you think it's not good, I don't like it, or I enjoy playing, or I learned something. Any feedback will help us make it better. And you need to respond, obviously, so that we know how to improve further. So this is a beta testing URL that's available here. So I will also post it on the YouTube channel for everybody to just copy it and go and register, and I'll send you the game links and how we would like to hear back from you. Now this concludes my presentation, and I would like to just tell you that I have described a single project today that I did, DPAPS. There are several other projects which are ongoing in the lab, and I have been working on genotype-phenotype correlations for a very long time, both in infectious and rare diseases. And I would like to, if anybody of you has interest in any of these domains, I would really like to talk to you offline. As Dr. Banerjee was saying that I work with a lot of kids, that's true. Most of my papers are with undergrad students or with master's students or with school students, and they're published in all international peer-reviewed journals. So point is that everybody has capacity to deliver and do science. I personally don't believe that degrees matter, because I have worked with some absolutely brilliant kids, and I'm showing two here. So the work that I presented today, DPAPS, on antibiotic peptides, the joint first author of this paper, Pooja, she got this paper when she was finishing her 12th standard. So she was just in school. She was very, very young. And then she went on to become the Gates Fellow, and now she's doing her PhD in cryo-electron microscopy from UK. The other fellow who came even earlier to my lab, Ashma, she is now a full scholarship fellow at Johns Hopkins University. So my experience with school students is extremely rewarding. They are just so energetic, and they read and they learn and they understand. Believe me, they can deliver sometimes much better than your PhD students can. So, and as is important, these are my lab members here in India and in France. And I would really like to thank all the open source community. I'm part of OSTD, Joggle, Lectures Without Borders. And I would like to thank funding agencies, CRI, CSIR, SERV, Gen2Pen Project, Hunter Trust for financing all this project over the years. And this is the campus. Hopefully the pandemic slows down and all of you would have a chance to come and visit. And with that, I would like to thank you. And if there are any questions, I'll be happy to take them.
Hi, can I ask a question, Dr. Banerjee?
Yes, please. Yes, please.
Yeah, so Dr. Bhardwaj, a really excellent talk. I really enjoyed it. So I'm going to be a little bit selfish here. I was wondering if you can direct your machine learning approaches to find, let's say, antimicrobial peptides for individual bacteria, because we have been studying antimicrobial peptides for Bordetella pertussis. We have a manuscript in preparation, and we find that when they form biofilms, they become resistant to antimicrobial peptides. So biofilms are extremely resistant to antimicrobial peptides, not surprising. But since we have a mouse model for Bordetella pertussis infection, I was wondering, can we target specific antimicrobial peptides, test their activity in vitro, and then start this as an adjunct therapy with vaccines to treat Bordetella pertussis infection? So if I were to go about that, which peptides would I be choosing or study?
Yes, so thanks for that very, very important question. See, when we started with this project, and I showed you the list of peptides initially we were looking at, so there were like 800 anti-biofilm peptides. And we were very excited about the fact that they were reported in approximately 25 gram-negative and gram-positive pathogens. So initially our objective was to develop an organism-specific machine learning model. But the data set was just too small to say anything. It would have produced only garbage. So we have then scrapped the idea and thought, okay, let us do a broad spectrum anti-biofilm peptide prediction for now, because there was not enough data. If you have enough peptides in Bordetella against which you have in vitro data of activity, there can be two types of models that can be generated. One, which classifies them into whether it's going to be active or not active. Second is a regression-based model where you can actually predict activity based on QSAR. You can actually say, okay, this is going to be probably the in vitro activity of this peptide in Bordetella. So both are possible depending on how much data set you have. And if you have data sets, we'll be very happy to generate new models in addition to running the peptides on our existing models and give you some prioritized set to test out. We'll be very happy to do that if you would like. And then the second thing is, if you have peptides that are not showing activity in Bordetella models, there is a way to generate mutants online currently. So first things first, what I would recommend is, if you have peptides that you know are active, and if our models also predict them to be active, there is some confidence with you that their models work. So it's first thing first is to test the models. If what you see, the model is giving something completely different, then it means it's not going in the right direction. So that's the first thing I would like to even do a feasibility check, eliminate or even use these models. And then second, if it is any useful, we can improvise them and make it more custom for Bordetella, and then take it forward.
Yeah, I will. Let's get in touch. I will send you an email and we can talk further by email or by Zoom. Thank you. I really enjoyed your talk.
Thank you so much, Dr. Dora.
Thank you, Dr. Angshu. It was really very, very interesting. I always get inspired from your talk when I see the end that the school kids are publishing. I feel so, because science really have no age. And it is very important that as a mentor, we are excelling or not. So I'm always inspired by your talk at the end. Well, so one of the questions that I see here is by Alex Echeverria. As in the tower defense game, does exist research on protective compounds that prevents biofilm formation instead to try to destroy it after formation?
Okay, so right now in the tower defense game at one level, we show that bacteria are moving in groups. So they are moving in a bubble. And if you continue to use the same antibiotics you were using to treat planktonic versions, they stop working at this level. So if you continue to use them, you are going to lose the game level. So at this point, we introduce something which is like a different version of a tower, which looks like a bulleted, gadgeted thing. So it's like a gamification of a different antibiotic. And that actually first disintegrates this bubble, and then the other antibiotic starts to work. So currently there is no provision in the game to, what should I say, protect a biofilm. Right now, the objective of the game is to tell the player that different antibiotics work in different fashion, and they have to be followed as is prescribed in the right duration, not less, not more. If there is a difference in terms of, you are prescribing this antibiotic to treat a biofilm, it's not going to work. So you need to first break open the biofilm, and then your previous antibiotic might still work. So the challenge with this thing is, when we did this play test with 14-year-old French kids, they were like, what the hell is a bacteria? And that is when we knew, okay, that's our first problem. Let us go simple. So we actually are trying to now think like a 12-year, 13-year-old child, and trying to, develop those concepts from a very basic thing onward, so that they actually, this is called, in gaming terms, it is called onboarding. So you're trying to first get them on board. And then the objective is to explain them different mechanisms of resistance and let the kids decide how the game should look like. They are recommending, like I have already spoken to a few kids, and they are recommending, why not in the next level you do this? And they are not just then talking the tower defense genre. They are basically expanding the genre of the game mechanics completely, which is extremely cool to watch. So yeah, it's a very, very interesting project because it's fun to do. It's extremely hard to implement, so it's a challenge on its own. But yeah, it's a huge learning for me and my team.
Excellent. Dr. Angshu, I have one last question from my side. When you were showing the biofilm formation, it's just for curiosity that initially I didn't have this idea, because initially the colonies of the bacteria, the planktonic bacteria, was like single colony. So two different colonies in the picture was separate. So with time, when they were like dividing, they came together and it became a mixed colony. So what basically, my question is, all these bacteria that are inside this biofilm has some symbiotic activity, not even symbiotic, they help the growth of each other, or even they can have antagonistic activity, because many bacteria with one another have antagonistic activity. So can they form biofilm or they cannot, something like that?
That's an amazing question. See, I was even very, very keen on understanding this, and that is when I came across papers which say that in a microcolony in a biofilm, the relationship between different bacterial, fungal, or X species is a master-slave kind of a relationship. So it's not symbiosis in most cases. So there is a master strain that decides that extra polymerase substance, what is the nature of that substance, and that dictates how the other microbes within that colony are supposed to behave. I've not seen many papers like that, but there are a few papers which indicates towards this kind of a colony architecture, which is a master-slave relationship. There are not many studies, because most people study biofilms in vitro, which are pure X species and not polymicrobial. So we don't have enough evidence to show what can grow together and what cannot really grow together. But yeah, I think that's a pretty interesting research project to identify what could be, or predict what could be, the potential compositions of biofilms, because that will allow us to show what kind of different infections they can cause at the same time. So it has a huge healthcare impact, I agree with you, but I am not aware of much data in that direction. This is all what I have read so far.
This is interesting actually, because I never thought that there can be something like master strain. It's like just a society like us, we human are living. It's just like the colony of animals. Please share with me this paper later on. I will be happy to read that. Thank you so much, Dr. Angshu. So we are going just probably 2 minutes late. So we will take a quick 10 minutes break to come back, just to have a coffee or something. So we will be back in 10 minutes for the next two presentations.
Thank you.
Buenos días nuevamente. Estamos regresando de nuestro break. Espero que hayan disfrutado su cafecito. A continuación, el próximo presentador es el doctor Felipe Gordillo. Él es licenciado en Bioquímica y bioquímico de la Pontificia Universidad Católica de Chile. Es doctor en Ciencias con mención en Microbiología de la Universidad de Chile. Hizo un postdoctorado en el Instituto Leibniz para Neurobiología en Alemania. Desde el año 2012 trabaja en la Universidad Católica del Maule en la implementación de la carrera de Ingeniería en Biotecnología, impartiendo clases y desarrollando investigaciones en las líneas de interacción planta-microorganismos, producción de aromas por levaduras y manejo de residuos de la industria de los alimentos, proponiendo soluciones amigables con el medio ambiente. Su trabajo se enfoca principalmente en el uso de hongos y bacterias aislados desde diversos sustratos naturales para la degradación de residuos agrícolas y la producción de biomoléculas de interés biotecnológico. Bienvenido, doctor Gordillo.
Muchas gracias, Alex. ¿Se escucha bien?
Sí, se escucha bien.
Ya, perfecto. ¿Se está proyectando?
Sí, está proyectando.
Bueno, gracias por la presentación, Alex. El título de esta presentación es Residuos industriales, una alternativa para producir biomoléculas de interés biotecnológico, que es una de las líneas de investigación que nosotros desarrollamos en el Laboratorio de Microbiología Aplicada, en el Centro de Biotecnología de los Recursos Naturales de la Universidad Católica del Maule. Bueno, ¿qué son los residuos industriales? O del inglés Food Manufacturing Wastes. En Chile aproximadamente se producen 1.5 millones de toneladas de residuos industriales al año, en donde aproximadamente el 45% se concentra en la región del Maule, Chile. Estos residuos industriales se componen principalmente de piel, semillas y pulpa del fruto. Y en esta región del Maule existen principalmente 3 empresas que producen estos tipos de residuos, que los generan al medio ambiente y generan, obviamente, contaminación ambiental. Estas son las industrias del vino, del tomate y del aceite de oliva. Los que generan, respectivamente, los residuos de o masa de la uva, o masa del tomate y alperujo, respectivamente. Ahí ustedes pueden ver en la fotografía esos tipos de residuos que se generan por estas empresas y la necesidad entonces de abordar estas con alternativas microbiológicas, las posibles contaminaciones ambientales que se generen y obviamente darle valor a estos productos que se generan posiblemente con el uso entonces de algún tipo de microorganismo que pueda degradar o atacar a estos sustratos. ¿Ya? Bueno, ¿qué son básicamente los residuos industriales? ¿Ya? Ahí ustedes ven una fotito de la composición de estos residuos industriales. Como son derivados de las plantas, del material celular, vegetal, estos residuos industriales, en su mayoría, ¿cierto?, que también reciben el nombre de residuos lignocelulósicos, se componen principalmente de 3 tipos de polímeros, como está graficado en esa figura: lignina, hemicelulosa y celulosa, los que están presentes en la pared celular de las plantas, como se ve en la figura. La celulosa, que está ahí abajito, acá, se compone aproximadamente en estos residuos entre un 40 a 60%. La hemicelulosa corresponde entre un 20 y un 40%, y la lignina corresponde al 10 al 25%. Fíjense cómo es la estructura de esos polímeros, muy importante. La celulosa es un polímero que está presente en fibras y está rodeado por un polímero de hemicelulosa y a su vez una especie de protección. y a su vez protegido también por estas fibras largas de lignina. ¿Ya? Estas fibras largas de lignina le dan entonces una rigidez en particular, y la diferente composición de estos polímeros le da diferentes propiedades a estos residuos. Por lo tanto, los residuos de alperujo, industria agroalimentaria, del tomate y del agua van a tener distintas composiciones y distintas degradabilidades según la variación en la composición de estos polímeros. Principalmente la celulosa y la hemicelulosa son polímeros fácilmente, entre comillas, degradables, porque básicamente funcionan a través de hidrólisis, mientras que la lignina es un poquito más recalcitrante a la degradación. Y básicamente estos tres polímeros son muy importantes en hoy en día en la generación de alternativas como generación de biocombustible a partir de estos tipos de residuos. Por eso la importancia de empezar a estudiar y valorizar estos tipos de residuos. El polímero central que estaba en la presentación, en la diapositiva anterior, que es el polímero de celulosa, que se ve más o menos en esta diapositiva, como les comentaba, es fácilmente degradable por microorganismos. Corresponde a un polímero de glucosa unido por enlaces del tipo beta 1,4, como se ve en la figura. Estos son unidades monoméricas de glucosa unidas por enlaces del tipo beta 1,4, glucosa. Esta celulosa es degradada por un montón de, una variedad muy grande de hongos y bacterias que están presentes en el medio ambiente. Y a través de hidrólisis pueden atacar este polímero y degradarlo a unidades más sencillas, como la glucosa. Los sistemas enzimáticos que tienen los microorganismos se componen principalmente de endoglucanasas, como está visto ahí, que son enzimas que rompen internamente el polímero, exoglucanasas, que van cortando el polímero por los extremos, y estas dos enzimas en su conjunto trabajan para generar oligosacáridos cortos, como está mostrado ahí, de 3, por ejemplo, unidades o de 2 unidades. A su vez se secretan al medio de cultivo por estos microorganismos otras enzimas que van acortando, obviamente, el polímero hasta la unidad fundamental, que sería la glucosa. Esas enzimas son la celodextrinasa y principalmente la beta-glucosidasa. Entonces el conjunto de acción de estas enzimas finalmente va a llevar entonces a la generación de la glucosa. Y la glucosa es utilizada como fuente de carbono y energía por estos mismos microorganismos. ¿Ya? Entonces los microorganismos cuentan entonces con baterías enzimáticas que en su conjunto trabajan entonces para degradar fácilmente, entre comillas, este polímero. Como modelo de estudio nosotros estamos trabajando con un hongo que se llama tricoderma para abordar entonces la degradación de estos tipos de residuos. Este hongo tricoderma fue aislado desde cuescos de aceituna en descomposición. Entonces de ahí entonces la intuición de que porque estaba presente en cuescos de aceituna podría entonces degradar, por ejemplo, los residuos industriales. Este hongo, como se muestra ahí, fue aislado en placas de agar papa. Por ejemplo, la figura 1A muestra el aislamiento, vale decir, uno tiene que sembrar una muestra y empezar a aislar hongos, porque crecen muchísimos hongos en estos medios. Y ahí en la figura A, en el panel A, se muestra entonces el hongo crecido en agar papa dextrosa, básicamente purificado, un cultivo de tipo exénico. También empezamos a caracterizar este hongo utilizando técnicas de microscopía óptica, como se ve en los paneles 1B y C. Se puede observar la morfología, ahí indicando con flecha las estructuras reproductivas de este hongo. Y una vez que uno separa estas estructuras reproductivas en un medio determinado, en el panel C ustedes pueden ver los conidios, que son estructuras reproductivas. Por lo tanto, tanto estas figuras 1A, B y C nos empezaron a mostrar, a evidenciar entonces que esta morfología del tricoderma correspondía básicamente al género tricoderma. Entonces, una forma de empezar a caracterizar estos hongos desconocidos es a través de técnicas microscópicas y observaciones en el microscopio. Lo siguiente que nosotros hicimos es seguir confirmando. Obviamente son las técnicas anteriores que son útiles, pero no son del todo certeras para empezar a tener más información sobre lo que realmente con lo que uno está trabajando. Entonces se abordó el análisis molecular de este hongo. Entonces se extrajo DNA cromosomal de este hongo y se amplificó la región ITS con partidores universales. Y utilizando la técnica de PCR, el producto amplificado se aisló y se secuenció. Eso se ve en el panel A. Se ve el producto de aproximadamente el producto esperado que corresponde al amplicón. Entonces ese producto se secuenció y en el panel B se está mostrando un análisis entonces de árboles de distancia en los cuales puede haber entregado esa información, uno lo compara con otras tricodermas que están presentes en la base de datos. Entonces los resultados, utilizando un análisis de neighbor joining con el parámetro Kimura, básicamente, mostraron que la secuencia amplificada, mostrada anteriormente, correspondería entonces a la de un hongo, que corresponde entonces al género tricoderma. Es decir, tiene una cercanía genética con, en ese caso, si lo ven ahí arriba, con tricoderma estéril. Entonces, con la información anterior y con estos análisis moleculares, uno está un poquito más seguro de que, por lo menos, este hongo aislado, que se va a probar con los residuos industriales, entonces pertenecería, entonces, por lo menos al género tricoderma. Entonces, lo siguiente que hicimos fue crecer este hongo a través, enfrentarlo a los residuos industriales a través de ensayos de crecimiento en placa. ¿Vale? Es decir, ustedes lo que hacen es colocar estos residuos en una placa de agar a una determinada concentración, inoculan el hongo purificado obviamente en el centro y empiezan a analizar su comportamiento, vale decir, su crecimiento en estos medios de cultivo. Esos son ensayos de placa, utilizando entonces los residuos al 0,5% como fuente de carbono y energía. Básicamente entonces esto para comparar el crecimiento del hongo en esos medios y utilizando glucosa como control. ¿Glucosa por qué? Porque glucosa en esos casos es una fuente de carbono bien asimilable. Básicamente todos los organismos heterótrofos crecen en estas condiciones y entonces podemos utilizarlo como control. Los residuos son complejos, la glucosa una molécula simple. Y además tiene otro comportamiento molecular que se lo voy a explicar más rato, que inhibe la expresión de las enzimas que a nosotros nos interesa en este caso, que son las enzimas que forman parte del sistema celulolítico de los hongos y de las bacterias. Y ahí ustedes pueden ver entonces que en el tiempo se van generando radios de crecimiento y muestra básicamente que el hongo es capaz de enfrentar y degradar y crecer en esos residuos y utilizarlos como fuente de carbono y energía. El residuo de pomasa del tomate que se ve en estas barras diagonales, se ve que está creciendo, pero fíjese que es un poquito más bajo con respecto a los otros dos residuos, a los residuos de la industria del alperujo y la pomasa de la uva. Y en blanquito ustedes ven la glucosa como control. Posiblemente la baja de crecimiento en este tipo de residuo, es decir, en el residuo del tomate, se deba principalmente a compuestos que se van produciendo y que puedan tener un comportamiento inhibitorio con respecto al crecimiento del hongo. Entonces, con estos experimentos preliminares, nosotros podemos decir que básicamente el hongo puede crecer en los residuos industriales. Lo siguiente que hicimos es también enfrentar este residuo, este hongo en los residuos utilizando medios de cultivo líquidos. ¿Ya? También se pueden utilizar, aparte de los medios sólidos, medios líquidos. Y en condiciones estériles, obviamente, uno puede inocular el hongo y analizar entonces el comportamiento que tiene, obviamente, ese hongo en esa fuente de carbono. La figura A, en el panel A, es básicamente un análisis preliminar que uno hace para testear rápidamente si el hongo produce enzimas de interés biotecnológico, en este caso las celulasas, en un medio. En este caso, esa placa contenía carboximetilcelulosa. Por lo tanto, uno puede visualizar que con técnicas especiales que se están produciendo las enzimas necesarias para degradar las enzimas de naturaleza celulásica. No sé si se puede ver un halo blanco que se produce alrededor de la colonia. Este es el micelio del hongo, y alrededor un halo blanco que corresponde entonces a la degradación o al trabajo de las enzimas cuando degradan un sustrato que está presente en esas placas, que es carboximetilcelulosa. Y con una atención especial uno puede hacer un revelado entonces de las enzimas que se van produciendo en el medio extracelular. Entonces, eso es un primer indicio de que ese hongo estaba produciendo esas enzimas celulasas, ¿ya? Lo otro que nosotros hicimos es los medios de cultivo, en cultivo líquido, como les comentaba anteriormente. Extrajimos los sobrenadantes de cultivo utilizando los residuos como fuente de carbono y energía, como les decía, al 0,5% en matrices independientes, y como control glucosa, ¿ya? Y extrajimos los sobrenadantes de cultivo y analizamos las actividades, en este caso enzimáticas, de los sobrenadantes de cultivo. Y ahí ustedes pueden ver en la figura, en el panel B, que los sobrenadantes de cultivo presentan actividad enzimática celulásica total utilizando un ensayo de papel del filtro. Estas 3 barras corresponden a los sobrenadantes de cultivo del hongo tricoderma crecido en los residuos industriales. Fíjense que tiene muy bajita actividad, casi nula actividad celulásica cuando se examina el sobrenadante que viene del cultivo de glucosa, ¿ya? Entonces, el hongo, cuando crece en glucosa, no tiene necesidad de producir las enzimas, porque tiene una fuente de carbono que es fácilmente metabolizable. Y posiblemente esté inhibiendo la expresión de las enzimas de tipo celulásico. Pero sí tenemos entonces crecimiento y actividad, obviamente, en los medios de cultivo de los residuos industriales. Y en el panel C se analiza la actividad beta-glucosidásica. ¿Se acuerdan esa enzima que degradaba el dímero de glucosa y lo partía y generaba entonces las moléculas de glucosa que se internalizan al hongo como fuente de carbono y energía? Fíjense que también tiene actividades beta-glucosidásicas, estos sobrenadantes de cultivo. Entonces, y muy baja actividad en el sobrenadante de glucosa. Entonces, también ahí se da algo muy parecido con respecto a la actividad celulásica total. Entonces, lo positivo de esto es que el hongo crece en estos residuos y también produce enzimas que degradan a su vez estos residuos. Obviamente tiene que producir estas celulasas, entre otras, para empezar a degradar los hidratos de carbono a unidades más sencillas y por ende crecer. Lo otro que analizamos también fue como una técnica complementaria, utilizando estos mismos sobrenadantes de cultivo, utilizamos sismogramas. Estos sismogramas permiten hacer un rastreo de las enzimas mediante técnicas o sustratos fluorescentes que son fácilmente detectables utilizando luz ultravioleta. Entonces utilizamos también los mismos sobrenadantes de cultivo. Esta vez se concentran los sobrenadantes de cultivo y pudimos detectar entonces estas mismas actividades enzimáticas, ya pero de forma un poquito más individualizada. Entonces determinamos las actividades celulásicas en el panel A y en el panel B las actividades beta-glucosidásicas. Ahí arriba entonces están cada línea descrito entonces los respectivos sobrenadantes de cultivo que se utilizaron. Glucosa, alperujo, tomasa, pomasa del tomate y pomasa de la uva. Entonces, cada una de esas bandas indican la presencia entonces de actividades enzimáticas o de proteínas, básicamente enzimas que están presentes en los sobrenadantes de cultivo y que están degradando entonces el sustrato que se utiliza para estos fines. Panel A, las actividades celulásicas, y panel B, actividades beta-glucosidásicas. Y esto se realizó a distintos tiempos de cultivo, por eso dice 0, 24, 48, 72, 96 y 120 horas. De cultivo se extraen estos tolerantes y se realizan estos tipos de ensayos. Y fíjense que hay actividades muy importantes en el tiempo, en el caso de las actividades celulásicas. Fíjense que aparecen unas bandas de actividades enzimáticas y aparecen en algunos casos más bandas de actividad enzimática, lo que sugiere la presencia de isoenzimas, ¿ya? Y en el caso de los solventes de glucosa, muy baja actividad enzimática. En el caso de las actividades beta-glucosiásicas, fíjense que también un comportamiento muy similar. Sin embargo, también aparecen actividades beta-glucosiásicas en los controles, en este caso en los medios de glucosa. Por lo tanto, da a suponer de que estas enzimas están presentes también en estos tipos de medios de cultivo simples, como el caso de la glucosa. Lo otro que hicimos fue estudiar en profundidad, ¿cierto?, estos sobrenadantes de cultivo, y separamos las proteínas extracelulares, lo que se denomina exoproteoma de los microorganismos en estos medios de cultivo. Se producen, como dato, estos hongos producen muchísimas proteínas distintas a los medios de cultivo, como exoproteoma, a diferencia de las bacterias que producen un poquito menos cantidad. Por lo tanto, estos hongos se utilizan, por eso el potencial de estos hongos, por el hecho de producir muchísimas proteínas al medio extracelular, y entonces empezar a estudiar esto. Mediante electroforesis en geles de poligliamida denaturantes, se separaron estas proteínas de los tolerantes de cultivo del hongo crecido en los residuos y en glucosa. Y ahí, como se puede ver en cada línea, está glucosa, residuo del perujo, residuo de la pomasa del tomate y residuo entonces de la pomasa de la. Fíjense que, bueno, panel A son geles teñidos con azul de Kumasi, y panel B los mismos muestras teñidas con nitrato de plata. El nitrato de plata es un poquito más sensible que los Kumasi y nos permite diferenciar de mejor forma entonces esos perfiles proteicos que nos interesan seguir estudiando. Entonces, fíjense los patrones, ¿cierto?, de proteínas presentes en estos medios, y lo otro que los patrones son obviamente distintos. Obviamente que en glucosa, que es un medio simple, una fuente de carbono simple, va a tener distintos patrones que el crecimiento en medios complejos, en estos casos los residuos industriales. A su vez, entonces queríamos también examinar el comportamiento de los exoproteomas en estos medios de cultivo, comportamiento diferencial. Pero básicamente el interés nuestro era seguir profundizando y analizar entonces el contenido de esas muestras. Y para eso nosotros necesitamos equipamiento potente, equipamiento que se denomina cromatografía y análisis de espectrometría de masa, para analizar todas las proteínas presentes en esas determinadas muestras. Entonces, eso es lo que se hizo en colaboración con un investigador alemán, Dr. Thilo Kenne, de la Universidad de Mannheim. Eso es lo que les quiero mostrar a continuación, que básicamente no es el análisis completo de las proteínas extracelulares en esa tabla, sino que escogimos básicamente de ese análisis lo que me interesa en esta presentación, que son las celulases. Fíjense ustedes que se obtuvo, analizando individualmente las muestras de los solventes de glucosa y de los tres residuos, aproximadamente 300 proteínas, de las cuales entonces, como les comentaba, nos centramos entonces con las proteínas de tipo celulásica. Entonces obtuvimos una lista entonces de este tipo de proteínas. Si se acuerdan de la primera figura, que se dividían entonces en endoglucanasas, exoglucanasas y beta-glucosidasas. Y ahí está marcado entonces las distintas hits que nosotros encontramos en los sobrenadantes de glucosa. Fíjense que encontramos básicamente las beta-glucosidasas presentes en esos medios, lo que está de acuerdo con lo encontrado en los sismogramas y con la baja actividad que nosotros observamos en los medios de cultivo. Y en los residuos industriales que se ven ahí, el perujo, la pomasa del tomate y la pomasa de la uva, encontramos muchísimas enzimas, tanto endoglucanasas, exoglucanasas, como obviamente las beta-glucosidasas. Lo otro importante hacer notar es la cantidad obviamente de enzimas y isoenzimas, es decir, enzimas que trabajan, que cumplen las mismas funciones, son isoenzimas. Por lo tanto, hay redundancia de función. ¿Ya? Eso hay que hacer notar. No es solamente una enzima que trabaja hidrolizando estos polímeros, sino que hay un montón de enzimas que en su conjunto trabajan entonces para hidrolizar, en este caso, la celulasa presente en esos residuos complejos. Como dato también anexo que no se muestra en esta presentación, también nosotros encontramos en los solventes de cultivo hemicelulasas y posiblemente algunas enzimas que también degraden el polímero de lignina. Por lo tanto, eso abre la posibilidad a futuros estudios. Eso por una parte, con respecto a la utilización de la tricoderma, ¿cierto? Les conté que habíamos aislado la tricoderma, la empezamos a caracterizar morfológicamente y analizamos entonces su comportamiento con respecto a los residuos industriales. Entonces encontramos actividades enzimáticas y las caracterizamos. Ya sabemos entonces la ciencia cierta cuáles son las proteínas que están involucradas entonces en la degradación. Pero tricoderma es un hongo, si bien es un hongo mesófilo, las proteínas que se producen posiblemente en ciertas aplicaciones, de hecho se utilizan en ciertas aplicaciones industriales, trabajan a temperaturas aproximadamente 35-37 grados. Son hongos mesófilos, trabajan a temperatura también. Por lo tanto, en nuestro afán de buscar otras alternativas o otros enzimas o microorganismos en otros ambientes que tengan también características o potenciales biotecnológicos importantes, fue que nosotros empezamos a trabajar con muestras antárticas, donadas por un investigador, Alex González, de la Universidad de Los Ángeles. De la misma forma con la cual trabajamos con tricoderma, empezamos a trabajar con estas muestras complejas antárticas y las enfrentamos a los residuos. De esa forma nosotros pudimos aislar una bacteria, que se muestra ahí en el panel, una colonia de una bacteria aislada. Fíjense que esa colonia es bastante irregular en su forma, que también se caracterizó morfológicamente, y presenta entonces una colonia de forma irregular. Cuando uno toma esa muestra y la analiza al microscopio óptico, utilizando una tinción de Gram, pudimos ver que esta bacteria tiene una forma basilar y tenía azul en Gram, por lo tanto es una Gram positiva. Es el panel B. Y en el panel C quisimos profundizar con una técnica de microscopía electrónica para ver, para confirmar obviamente lo que se había visto con microscopía óptica. Y se confirmó entonces que la bacteria era basilar y además tenía la presencia, esta bacteria contenía también esporas. Por lo tanto, estas tres informaciones nos daban a pensar de que la bacteria podría pertenecer al género Bacillus. Pero al igual que en el caso de la tricoderma, nosotros teníamos que confirmar eso a través de técnicas moleculares. Entonces se hizo lo mismo que para tricoderma, se aisló el DNA cromosomal de esta bacteria y se amplificó la región 16S de este RNA ribosomal, utilizando partidores universales. Se amplificó esta secuencia utilizando PCR, se aisló, se secuenció y se enfrentó entonces a la base de datos y se comparó entonces con otros integrantes del género Bacillus y también se elaboró un árbol de distancia. Se muestra en el panel B. Los resultados utilizando las mismas técnicas de la secuencia amplificada correspondería entonces a la del género Bacillus, debido a la cercanía genética con varias bacterias del mismo género, como se muestra ahí arriba, ¿cierto? Tales como Bacillus mycoides, Bacillus pacificus, entre otros. Por lo tanto, igual que en el caso de la tricoderma, nuestra bacteria, en este caso, le asignamos entonces la pertenencia al género Bacillus. Entonces, tanto tricoderma como Bacillus, con estas técnicas moleculares, se podrían entonces caracterizar utilizando estas herramientas. Lo otro que hicimos entonces es algo similar que con respecto al hongo. En este caso, las bacterias son un poco más sencillas, ¿cierto? Tienen otras técnicas para empezar a estudiarlas. Por lo tanto, no utilizamos un crecimiento radial, sino que utilizamos directamente el conteo en matrices de cultivo. Entonces, lo que uno hace es enfrentar esta bacteria con los distintos residuos en un determinado medio de cultivo, en las mismas condiciones, teniendo glucosa como control y los tres residuos en forma separada. Manipulando entonces el hongo en esos medios estériles y observando el crecimiento. Y como se puede observar, esto obviamente sacando muestras a distintos tiempos y examinando la población microbiana como aumentaba en el tiempo. Entonces, si se fijan, todas las curvas de crecimiento son de forma exponencial con una fase estacionaria. Por lo tanto, nos da cuenta de que el hongo crece tanto en glucosa, obviamente. Glucosa es una fuente de carbono que es fácilmente metabolizable por la mayoría de los microorganismos, y en los residuos industriales. El residuo de la uva, del alperujo, de la industria de la oliva, ¿no? Y de la masa del tomate. Las B, C y D respectivamente. Entonces, eso es bastante positivo, ¿cierto? Que el hongo, que la bacteria pueda crecer en esos tipos de residuos. Por lo tanto, uno esperaría, por lógica, que para crecer en estos medios complejos, la bacteria en este caso tenga que producir al medio extracelular enzimas que degraden estos polímeros complejos para llevar las unidades monoméricas al citoplasma de la bacteria para su crecimiento. Entonces, la misma lógica, analizar entonces estas enzimas, ¿cierto? Las enzimas que se producen en los sobrenadantes de cultivo. En el panel A hicimos un análisis un poquito similar a lo que se utilizó para el hongo, con respecto a la tinción contra el hongo, en este caso una tinción con yoduro. Se fijan, es la bacteria sembrada en un medio en particular. En la zona izquierda, entonces está la bacteria sembrada, la bacteria antártica que nosotros aislamos, Bacillus, y en la parte derecha ustedes ven también como control, sembramos una bacteria también del género Bacillus, pero mesófila. No sé si logran ver un halo que se produce por fuera de esta bacteria antártica, que correspondería a la secreción o a la actividad enzimática intracelular que es producida entonces por esta bacteria antártica, ¿ya? No así el caso de esta bacteria mesófila, que al parecer carece entonces de estas enzimas para degradar estos polímeros complejos. Ese es un análisis bastante cualitativo entonces que uno puede hacer, igual como el caso de la tricoderma, para estudiar muy rápidamente entonces la presencia de estas enzimas. Y similarmente, entonces, como se realizó en el caso de la tricoderma, nosotros aislamos los sobrenadantes de cultivo y enfrentamos esos sobrenadantes de cultivo en ensayos de actividades enzimáticas, obviamente tanto la celulásica, que se realizaba utilizando el papel del filtro, y los ensayos de beta-glucosidasa. Encontramos, para sorpresa, actividades obviamente esperables en todos los medios de cultivo, alperujo, pongas el tomate, uva, comparando obviamente con el control de glucosa. Encontramos, para nuestra sorpresa, actividades también en glucosa, ¿ya? Y los niveles de actividad son un poquito más bajos que con respecto a la tricoderma. Acuérdense que tricoderma, según esa tabla, produce un montón de enzimas que actuarían entonces en la degradación compleja de estos residuos. Les comentaba también que el caso de las bacterias son mucho más sencillas y posiblemente produzcan menos cantidad. Por ende, los niveles de actividad enzimática tenderían a ser un poquito más bajos si los comparamos con lo obtenido con hongos. Y para sorpresa nuestra, también analizamos las actividades no solamente a 30 grados, como en el caso analizado para tricoderma, sino que también a 50 grados Celsius. Y a 4 grados. O sea, uno esperaría por lógica, si que es una bacteria antártica, tiene que sobrevivir en esas condiciones ambientales de temperatura muy baja. Por lo tanto, obviamente, producir enzimas en esas condiciones. Por lo tanto, esa fue un poco la lógica de analizar las actividades enzimáticas a esas temperaturas. Como les comentaba, para sorpresa, encontramos actividades enzimáticas cuando hacemos los ensayos a 4 grados Celsius. Entonces, eso es muy positivo porque estas enzimas, hay ciertos procesos que requieren baja temperatura y podrían entonces aplicarse este tipo de enzimas esos procesos que requieren baja temperatura. También alta temperatura, como se ve, 50 grados Celsius. Entonces, encontramos tanto actividades celulásicas totales, que corresponderían a una mezcla de las actividades endoglucanasa y exoglucanasa, y actividades beta-glucosidásicas en los sobrenadentes de cultivo cuando uno crece esta bacteria en los residuos industriales. Como conclusiones de nuestro trabajo, ahí tienen ustedes que tanto el hongo como la bacteria antártica Bacillus sp crecen sobre los residuos industriales de la región del Maule, produciendo celulasas que podrían tener un potencial biotecnológico, como por ejemplo la generación de biocombustibles, donde sí se utilizan como manera alternativa estas enzimas para degradar esto, generar moléculas de glucosa que las toman otros microorganismos como las levaduras para producir, por ejemplo, bioetanol. Futuras secuenciaciones del genoma de estos microorganismos nos podrían ayudar a entender el rol de estas enzimas y otras en los procesos de degradación y obtención de biomoléculas con alto potencial biotecnológico. Yo quiero también este trabajo agradecer a las personas que hicieron posible, que es el colaborador, Dr. Thilo Kenne, de la Universidad von Gerig Universitat de Magdeburg, que nos ayudó con la secuenciación de las sobrenantes de cultivo, y eso es lo que nosotros queremos hacer, obviamente aplicar para el caso de la bacteria antártica, analizar el exoproteoma en esas condiciones de cultivo para obviamente validar el hecho de que nosotros encontremos celulasas en esos medios de cultivo. Y también agradecer a todas las personas que hicieron posible este trabajo. Son los estudiantes de la carrera de Ingeniería en Biotecnología de la Universidad Católica del Maule, a la señorita Claudia Faúndez, Gloria Guerrero y Sofía Díaz. Muchísimas gracias.
Muchas gracias, doctor Gordillo, por su presentación. Tenemos tiempo para un par de preguntas. Estoy viendo en el chat que la doctora Banerjee pregunta: ¿los residuos agrícolas lignocelulíticos
Lignocelulósicos
también se utilizan para producir EPS bioactivo? ¿Cuáles son los factores que debemos controlar para usar residuos lignocelulíticos para la producción comercial?
Bueno, esto es a nivel de laboratorio, pero a nivel industrial obviamente que se requieren o se utilizan básicamente se están empezando a utilizar estos residuos utilizando diversos tipos de hidrólisis química, hidrólisis por vapor y otra alternativa que se está proponiendo y que se ha trabajado hace mucho tiempo atrás también con el caso de otros tipos de residuos, son la presencia de estas enzimas que actúen degradando estos residuos y generando obviamente los monosacáridos, en este caso por ejemplo carbohidratos, para que sean tomados por otro organismo que produce, en este caso, por ejemplo, bioetanol, o algún producto que se quiera utilizar, que se quiera también producir de forma secundaria. Eso va a depender del microorganismo secundario. Pero la idea principal es inicialmente ensayar estas pool de enzimas en estos residuos, pero en condiciones ya más industriales, ¿cierto? Entonces, básicamente nosotros estamos preliminarmente analizando la presencia de estas enzimas utilizando las técnicas que disponemos. No sé si respondí.
Otra pregunta, doctor Gordillo. ¿Probaste la actividad enzimática a temperaturas bajas para el bacilos? Y para tricoderma, ¿lo hiciste? ¿Cómo es comparativamente la actividad enzimática de tricoderma comparada con bacilo? A temperaturas bajas.
Lo que se mostró, o nuestro trabajo mostró, es el análisis a temperaturas de 30 grados Celsius para el caso del hongo. No hemos analizado actividades enzimáticas tanto a 4 grados como a 50 grados Celsius. Es una cosa que nosotros queremos hacer, porque está pendiente para hacer básicamente la comparación con lo que se encontró en bacilos. Pero como les comentaba en la presentación, las actividades enzimáticas son más bajas, obviamente, porque los hongos producen muchísimo más enzimas al medio extracelular. Por lo tanto, esas actividades están dadas por la presencia de todas esas isoenzimas que están presentes en el medio extracelular. Suponemos que Bacillus, como dato, los hongos aproximadamente producen por sobre 300 proteínas distintas en los ordenantes de cultivo. Las bacterias producen aproximadamente 100 proteínas. Entonces, de esas 100, obviamente que hay que sacar las proteínas de tipo celulasa. Entonces, nosotros esperamos con los análisis de los exoproteomas encontrar posiblemente muy baja cantidad con respecto a tricoderma de las enzimas celulásicas, de las endo, la exo y la beta glucosías. Generalmente se producen 1, 2 o 3 tipos de proteínas. Por lo tanto, eso da cuenta de la baja actividad cinética que nosotros encontramos. Y lo compara con el hongo.
Perfecto. Muchas gracias, Felipe. Doctora Banerjee, le doy la palabra.
Creo que hay otra pregunta también para Felipe.
Perdón. Alexander Galán pregunta, considerando los grandes volúmenes y las altas tasas de producción de los residuos de las industrias utilizadas para el estudio, ¿qué tan realista es usar estos microorganismos para degradarlos o reciclarlos?
Sí, buena pregunta. Eso yo creo que la persona más idónea sería un ingeniero que nos podría ayudar, ¿cierto?, en futuros proyectos para analizar la factibilidad de utilizar esto o de encadenar esta información, ¿cierto?, y llevarla a los respectivos centros de acopio de estos residuos, ¿cierto? Si bien se producen muchísima cantidad de residuos, la competencia a estos tipos de procedimientos obviamente que son otras alternativas como los compostajes. Pero sí se podría destinar obviamente una baja cantidad de estos materiales para la producción de estos tipos de enzimas a nivel de escala muy reducida, muy baja. Entonces, la factibilidad no me atrevo a responder porque no sé muy bien cómo funciona a nivel industrial este tipo de escalamiento, pero solamente decir que se podría obviamente realizar un análisis para ver la factibilidad a través de escalamiento.
Ok. Muchas gracias, doctor Gordillo. Ahora sí cedo la palabra a la doctora Banerjee.
Bueno, a la presentación final del día 1 estará a cargo del doctor Alex Echeverría. Tenemos doctor Echeverría ahora con nosotros, ahí. Doctor Echeverría es doctor en Ciencias Aplicadas con mención Sistemas Marinos Costeros de la Universidad de Antofagasta, Chile. Licenciado en Ciencias Biológicas, biólogo con mención en Bioprocesos y bachelor en Ciencias. El autor de 17 publicaciones indexadas, con 18 años de experiencia y participación en más de 20 proyectos de investigación y desarrollo en las áreas de biotecnología, ecología microbiana, fagoterapia y biominería. Actualmente trabaja como académico investigador adjunto en la Universidad Católica del Maule, del Talca, Chile. Doctor Echeverría, por favor.
Hola de nuevo. Yo le vengo a contar una historia. Las bacterias, como dijo un querido profesor de la universidad, son organismos maravillosos. Son organismos superpoderosos, capaces de sobrevivir y de adaptarse a los ambientes más extremos presentes tanto en este planeta como en quién sabe cuál más. Como dominio o como reino, las bacterias son resilientes, prácticamente inmunes a cualquier cambio ambiental, incluido cambio climático. Incluso tienden a estabilizar sus efectos, para que el resto de los habitantes de la Tierra, como nosotros, lo podamos soportar de mejor forma. El problema para nosotros ocurre cuando su capacidad de mantener este equilibrio se ve superada por la fuerza del impacto. Bueno, en esta ocasión les voy a presentar un ejemplo de cómo el cambio climático puede llegar a impactar la microbiología de los lagos andinos. El título de mi presentación es El cambio climático e impacto en la microbiología de lagos andinos, y yo soy el doctor Alex Echeverría. Acerca de los lagos de los que le voy a hablar, el señor Benjamín Vicuña Maquena, en el año 1873, cuando fue intendente de Santiago, lo describió de esta manera: una silenciosa belleza del lago, terso espejo que apenas si el suave ambiente de la tarde rizara de cuando en cuando, haciendo girar en su nítida superficie brillantes crespones de espuma que no conseguirían borrar de su seno el retrato de los acantilados picachos y atrevidas escarpas que le sirven de marco. Esa es la descripción del lugar que les voy a mostrar ahora. O sea, ya se pueden imaginar la belleza descrita tan poéticamente por don Benjamín Vicuña Maquena en ese tiempo. Y actualmente yo no cambiaría nada de esa descripción. La información del lugar es esta. Es una laguna, o un lago, perdón, dos lagos, que se llaman Laguna Negra y Lo Encañado. Son dos lagos que pertenecen al mismo sistema lacustre. Son oligotróficos y monomícticos. Y las partes que se pueden observar acá, bueno, están en la cordillera de la región metropolitana a 2.700 metros de altura, que eso también es un dato importante. Estos dos lagos están alimentados por un par de cuencas que provienen o que están coronadas por el glacial Echaurren, que se ve acá arriba. Y en esta figura se muestran los ingresos de las cuencas, o por dónde entra el agua a cada una de los dos lagos. En Laguna Negra, la entrada es por acá, y se observan esos puntos que son dos pozas de retención, que son muy importantes en este estudio, y es bueno que las tengan en cuenta. Esta de acá también es otra poza de retención que viene desde esta entrada de glaciar. El lago Lo Encañado, alguna vez estuvo conectado a Laguna Negra, actualmente está totalmente desconectado y recibe agua netamente del deshielo glacial a través de esta cuenca, en donde el curso es directo sin pozas de retención. Un corte transversal del lugar es como se muestra. La Laguna Negra está a 2.700 metros sobre el nivel del mar, lo encañado a 2.492, lo que lo hace 300 metros, 200 metros más bajo. Originalmente hubo un influjo de agua desde aquí hasta acá, que se perdió. Ahora algo de influjo queda a través de algunas vertientes, pero por las cuales el agua pasa filtrada a través de la roca, ya no es directamente desde una laguna hacia la otra. Acá arriba el esquema muestra el glacial Echaurren, que alimenta los dos lagos. Esto es un dato también muy importante. Cómo ha variado a través del tiempo estos tres factores que estoy graficando acá. En primer lugar, la temperatura. La temperatura es esta curva roja, en donde se grafican los promedios anuales de temperatura, y se observa en la línea punteada la tendencia de esta temperatura a lo largo de los años, desde el año 1991 hasta el año 2012. Se nota un incremento en las temperaturas promedio del lugar. Estos datos fueron obtenidos desde una estación que se encuentra en el Cajón del Maipo, a cerca de 3 kilómetros de la laguna, ¿no? Las precipitaciones, en azul, están acá, y se observa que a través del tiempo, como seguramente ya lo habrán notado, en Chile, hay un descenso de la cantidad de precipitaciones que ocurren. Las precipitaciones están graficadas aquí en el eje de la derecha. La pantalla se come un poquitito el nombre, pero son milímetros, ¿no? Y en tercer lugar, en la línea verde, es el balance de masa del glacial Echaurren, que nos está mostrando a través de su línea de tendencia que el glacial está perdiendo masa a través de los años. Ha perdido una buena cantidad de masa que está expresada en kilogramos por metro cuadrado por año. Eso quiere decir que el glacial está retrocediendo, dado el impacto del cambio climático. Y este impacto se puede ver acá, en un simple ejercicio que usa Google Earth y que muestra el área de cobertura del glacial Echaurren en el año 2004, en la foto de la izquierda, y en el año 2014, en la foto de la derecha. Se observa claramente que el glacial ha retrocedido un buen trecho, y se nota además el cambio en el volumen de la laguna, o en el área de cobertura del lago, que se llama Laguna Negra, a través del tiempo. Aquí esta sección que se ve acá es la unión que existía antiguamente entre la Laguna Negra y el lago Lo Encañado. Esta zona, como se ve en el año 2014, ya no existe. Entonces ya no hay una comunicación a través de un canal. Además, las pozas de retención que les mostré anteriormente se generaron aquí, antes no existía, y Lo Encañado continuó recibiendo aporte directo del glacial por esta cuenca, y Laguna Negra lo recibe primero en las pozas de retención y después cae al lago. Bueno, esta situación llamó la atención de la gente, de gente extranjera, principalmente de gente de la NASA. Y se generó un proyecto que se llama Planetary Lake Lander, en la que 3 equipos de investigación de varias partes del mundo, equipos multidisciplinarios, fueron financiados por NASA, y se generó un programa para estudiar un sistema de glacial asociado a una laguna, para probar, finalmente, su objetivo final era probar un sistema autónomo de exploración de ambientes líquidos en nuestro planeta, para futuras operaciones en la luna de Titán, en donde el ambiente es líquido y se sabe que no hay una parte sólida para aterrizar un robot como lo hacen en Marte. El Planetary Lake Lander es en el fondo un equipo, es un pontón, una especie de pequeña balsa que está lleno de sensores y obtiene datos críticos acerca de cómo el cambio climático y el retroceso de los glaciales afecta el hábitat, la limnología, la biodiversidad, los ciclos bioquímicos y el ecosistema en general. Y el sujeto de estudio fue principalmente la Laguna Negra, o el sistema Laguna Negra Lo Encañado, en la cordillera chilena. Estos son una muestra de los equipos que utilizaban. Estas cosas iban bajo el agua. Son sensores de distinto tipo, de clorofila, de temperatura, de pH, etc. Y estos sensores iban en este botecito, iban metidos a 2 o 3 metros de profundidad, iban midiendo constantemente, mandaban una señal a una antena satelital, que la derivaba a un satélite a Estados Unidos, y desde allá iban registrando los datos. Es una forma de probar que el sistema funcione para transmisión de datos a distancia. Esta parte la puso, obviamente, la NASA. Y la parte que pusimos nosotros fue esta. El equipo de muestreo químico y microbiológico, ¿no? Nosotros somos el equipo de muestreo químico y microbiológico en ese tiempo. Aquí estoy con un colega español, con Antonio, un colega chileno con el que fuimos a muestrear. Anduvimos en lancha, tirando botellas Niskin, con sensores multiparámetros, tomando muestras de agua, para determinar cómo la microbiología se cambiaba en estos dos lagos y cómo era, cuál era el efecto de la deglaciación, ¿no? Entonces, a grandes rasgos, la metodología microbiológica fue filtrar mucha agua, 100 litros de agua. Usamos filtradores tangenciales, que tuvimos que acarrear por la cordillera, ¿no? Para poder filtrar, transformar los 100 litros de agua en 250 ml para después poder filtrarlo y hacer extracción de ADN. Se extrajo, purificó, se secuenció por Illumina MySeq, se secuenció la región B1, B3, de varias muestras a distintas profundidades de ambos lagos, y se analizó por Kime 1.9 y Primer 6, utilizando Permanova para los análisis estadísticos. Una foto para que vean de más cerca cómo era el agua de Laguna Negra y por qué se llama Laguna Negra. El agua es súper transparente, súper prístina, se ve negra con el reflejo del cielo. Y como pueden ver, es tal como la describió Benjamín Vicuña Maquena, un espejo que refleja las montañas a su alrededor. Sus dimensiones son 6.100 por 1.700 metros y tiene 276 metros de profundidad. Y esta cosa amarilla que está aquí es el PLL, que le llaman los españoles, el PLL. Es el Planetary Lake Lander, que es el equipo con sensores. Laguna Lo Encañado. Es una laguna más pequeña. Ustedes pueden ver claramente aquí que el color del agua es distinto, es un tono más verdoso, dado por los sedimentos del glacial. Esto que se ve acá es la cuenca, perdón, acá no les mostré. Esta es la cuenca, este es el glacial Echaurren, esta es una de las bajadas, y esta es otra de las bajadas. Esto alguna vez fue cubierto de hielo, y ya no queda hielo prácticamente, y el glacial está retrocediendo hacia allá. En Lo Encañado, como les dije, el agua es distinta por los sedimentos arrastrados, porque no hay una poza de retención de sedimentos antes de llegar a la laguna, y el glacial Echaurren está por acá, y desde aquí alimenta esta laguna, o este lago. Las dimensiones son 980 metros por 635, con 45 metros de profundidad. Es un poco más pequeño, pero es bastante significativo en cuanto a tamaño. Lo primero que quiero mostrarles son los perfiles comparativos de ambos lagos. Perfiles de temperatura, la termoclina de este lago, se ve claramente en la línea roja que Lo Encañado tiene una mayor temperatura en la superficie, dado por el calentamiento producido por los rayos solares en la superficie, pero como la luz no penetra, como se ve acá, en este gráfico, la luz no penetra mucho en Lo Encañado, entonces esta temperatura baja abruptamente a partir de los 5 o 10 metros. Así también la turbidez aumenta en la zona donde la temperatura es más alta y disminuye rápidamente después, o no tan rápidamente, disminuye después, pero nunca llega a ser tan poco turbia o tan transparente como Laguna Negra, que está aquí. La clorofila. La clorofila tiene un aumento en la misma zona donde está el aumento de turbidez y la misma zona donde, hasta donde llega la penetración de luz. Hay que tomar un poco en cuenta que los organismos fotosintéticos probablemente se ubican en este sector porque es donde mejor le llega la radiación par y no le llega tanta radiación UV. Entonces se protegen un poco a cierta profundidad, y es aquí donde está el pic de estos organismos. La materia orgánica disuelta también, o la materia orgánica disponible, también en Laguna Negra es mucho menor que en lo encañado, y la conductividad lo mismo. La conductividad en lo encañado es más alta en la superficie que en la profundidad. Estos 4 diamantes que ven aquí en toda la figura son las 4 zonas de donde se tomaron muestras de agua y se analizaron. ¿Ya? Bueno, NTU son las unidades nepelométricas de turbidez, y RFU las unidades de fluorescencia relativa que se muestran en los gráficos, NTU y RFU. Las consecuencias de esto es que la incorporación del sedimento proveniente del glacial agrega nutrientes al sistema, disminuye la entrada de luz y genera refugio para ciertos microorganismos. Además, el índice de estado trófico, el TSI, sacado o medido a través de disco Secchi en lo encañado, es cercano a 30. O sea, es casi mesotrófico, por definición del índice de estado trófico. Versus 10 en Laguna Negra, que es oligotrófico. Elementos como el sodio, perdón, el calcio, el silicio y el magnesio se incrementan en lo encañado, además de la materia orgánica disuelta y la clorofila en la zona superficial, como vimos en los gráficos recién. El número total de microorganismos contados fue mayor en lo encañado, un orden de magnitud mayor, 10 elevado a 5 versus 10 elevado a 4. Y además se tiene una mayor diversidad y una menor dominancia en lo encañado, que quiere decir que tiene más especies. Perdón, se me fue para el otro lado. Que tiene más especies y está más equilibrado en cuanto a la cantidad de especies, no tiene tanta dominancia. Aquí les muestro un heatmap. No se confundan tratando de verlo completo, sino que más bien aquí arriba está el árbol, el dendrograma que compara las distintas lagunas L0, 5, 10, 20, 0, 5, 10, 20 significa las profundidades desde donde fue tomada el agua. Aquí se ve que hay una diferencia significativa entre los lagos respecto a la comunidad microbiana. Esto es distancia respecto a sus comunidades microbianas. Similaridad respecto a sus comunidades microbianas, por Bray Curtis. Sin embargo, no hay una diferencia clara y significativa respecto a las profundidades tomadas dentro de un mismo lago. Eso quiere decir que las comunidades microbianas son bastante homogéneas entre el 0 y el 20 metros de profundidad. Se observan algunas diferencias en cuanto a las bacterias que están presentes o que provocan esta diferencias de similitud entre uno y otro lago, pero quiero que se fijen un poco en las fototróficas, que aquí es donde está el tema más importante de este estudio, ¿no? Por ejemplo, las cloroflexi, las cianobacterias, tienen ciertas diferencias. Algunas alfa-protobacterias, beta-protobacterias, que también son algunas fototróficas. Las vamos a ver un poquito más detalladamente más adelante, y son las que principalmente marcan la diferencia entre uno y otro, dado su estado digamos, el metabolismo presente en uno y otro lago cambia. La relación con los factores ambientales, esto es un análisis canónico, son dos análisis canónicos, uno respecto a los factores físicos que existían ahí, medidos principalmente con el PLL, y otro con los factores químicos, que son análisis químicos del agua que se tomó, de las muestras de aguas tomadas de la laguna, ¿no? Se observa siempre la agrupación por lago. Aquí el LN20 se separa un poquitito, principalmente por la temperatura. Considerando el factor temperatura, el LN20, la temperatura es muy baja, entonces es uno de los factores que origina esta separación del grupo. No es tan relevante respecto a la comunidad microbiana, pero provoca la separación. Se ve, de nuevo, la clorofila A, el F2 y la turbidez es mayor en lo encañado, y la penetración de luz se carga hacia la laguna negra, ¿no? Y por el lado de los factores químicos, una mayor concentración prácticamente de todos los iones ocurren en lo encañado, dado el influjo glacial directo que tiene, que se ve también, uno podría predecirlo mirando solamente el color de la laguna, ¿no? Estos son dos NMDS, análisis multidimensionales no métricos, ¿no? Correlacionando con los filos que están presentes. Tenemos que en la Laguna Negra hay mayor cantidad de filos que tienen géneros o clases que son fototróficas. Eso es una cosa que a mí, en lo personal, me llamó la atención cuando lo vi por primera vez, porque yo me imaginé que por tener más nutrientes, había una mayor cantidad de fototrofía en lo encañado. Pero no fue así. La laguna o el lago que tenía agua más clara tenía más cantidad de microorganismos, de bacterias, pertenecientes a grupos fototróficos. Y aquí se ve en el mapita del lado claramente, si yo hice el trabajo de juntar todos los géneros y clases que pertenecían a grupos fototróficos y los comparé y los separé de los no fototróficos, los marqué, y después le pedí al programa que me dijera hacia qué lado se correlacionan los fototróficos y los no fototróficos, y la correlación es clarísima. Los no fototróficos están hacia el lado de lo encañado, y los organismos fototróficos están hacia el lado de la laguna negra. Ahora, de nuevo, los organismos fototróficos o fotosintéticos están aquí. Estos son los grupos de organismos que yo encontré. Son los clorobi, cloroflexi, cloroplastos, cianobacterias. Los cloroplastos también los incluí, los consideré importantes. Cianobacterias, alfa-protobacterias, beta-protobacterias y gamma-protobacterias. No quiere decir que toda la clase gamma-protobacteria sea fototrófico, sino que integrantes del grupo de gamma-protobacterias que tienen cierta fototrofía fueron incluidos acá. Lo mismo con los beta, con las alfa-protobacterias, y se ve que, por ejemplo, cloroflexi es uno de los microorganismos que más porcentaje de presencia tiene, sobre todo en Laguna Negra, hasta los 10 metros. En 20 metros ya disminuye. Las alfa-protobacterias que están en azul, son estos cuadros azul, también son un grupo importante. Asimismo, las beta-protobacterias, pero las beta-protobacterias son un poco más importantes en lo encañado respecto a su cantidad total que en Laguna Negra, ¿no? Entonces, como conclusión, el cambio climático global ha modificado notablemente la integridad de glaciales Chaurren, cambiando la dinámica de las cuencas de alimentación de ambos lagos. El hecho de que se hayan originado estas pozas de retención y que el influjo glacial haya disminuido drásticamente en Laguna Negra por el retroceso de glacial, que ahora alimenta mayormente a lo encañado que Laguna Negra, ha provocado un cambio grande en la dinámica de las cuencas de alimentación. Las comunidades bacterianas de ambos lagos son significativamente diferentes, debido principalmente a la conformación de sus cuencas, lo que a su vez determina la concentración de nutrientes y disponibilidad de la luz. O sea, de nuevo, el cambio que ocurrió en las cuencas determina los nutrientes que ingresan a cada uno de los lagos y la disponibilidad de luz que tienen cada uno de estos lagos. Una importante diferencia metabólica está dada principalmente entre microorganismos en los que predomina la fototrofía enoxigénica. Entonces tenemos entre los microorganismos que estaban allá, beta protobacteria, algunas alfa protobacterias, gamma protobacteria que también pueden producir fototrofía enoxigénica, actinobacterias y cloroflexi también, principalmente, que era uno de los organismos mayoritarios, son determinantes en la generación de oxígeno, ¿no? Bueno, son determinantes en la producción primaria de estas lagunas, pero no generan oxígeno. La acción directa de la entrada de sedimentos por deglaciación a un lago oligotrófico es la disminución de la fotosíntesis oxigénica, provocando una menor captación de CO2. Entonces, entran más nutrientes, pero eso no significa necesariamente que aumente la fotosíntesis oxigénica, sino que provoca una fotosíntesis muchas veces que es anoxigénica, entonces no permite captar el CO2 del ambiente, lo que al final se transforma en un círculo vicioso, porque se va generando más CO2 ambiental, se va captando menos mientras más se genera, y se va autoestimulando el calentamiento global. Bueno, la bibliografía principalmente, este trabajo está publicado en Frontiers el año 2018. Es este trabajo que está aquí, en conjunto con gente de distintas universidades y gente de la NASA, del Planetary Lake Lander, como ven. Y aquí está todo el equipo. No encontré las fotos de todo, así que preferí poner un listado. Y se nota bien aquí la cantidad de instituciones que están presentes y la cantidad de expertise que aporta cada uno a este proyecto. Oh, bueno, muchas gracias y estoy atento a las preguntas.
Dios mío, Alex, muy, muy bonita presentación. Cuando lo vi tu presentación, estuve pensando un tema. Cómo comunidad microbiana está cambiando en los lagos. No sé que si existe mismo tipo de cambio en aguas termales o no, porque en caso de lagos, no está como de millones de años, más reciente, formaciones más reciente comparación con aguas termales. Y si hay trabajo, no sé que si tú tienes idea o no, si hay trabajo en aguas termales así, por cambio de comunidad microbiana.
No tengo conocimiento acerca de trabajos en cambio de las comunidades microbianas en aguas termales. Tienes razón en lo que dices respecto al tiempo, las épocas en que se ve, o la gracia de esta investigación más bien, es el hecho de que todo ocurrió muy rápido. En 10 años tuvimos cambios sustanciales. Desde un momento en que los dos lagos tenían una comunidad bacteriana más o menos similar, el hecho de que cambiara el sistema, de que cambiara las cuencas, provocó que cambiara completamente la comunidad. Ahora, en aguas termales generalmente son un poco más estables, ¿no? En cuanto a su estado. Entonces, no sé si existe un trabajo respecto a eso.
Y otro, no sé, en nuestro Maule tenemos Laguna del Maule. Yo creo que no hay reportado de este tipo de cambio de comunidad en Laguna del Maule también. Y Laguna del Maule, esta región es como fuente de todas las aguas termales en región del Maule, porque creo que esta fue un volcán millones de años antes para una explosión geotérmico y probablemente un área súper interesante para pensar por nuestro
Sí, la Laguna del Maule también está en la mira de la gente de la NASA, ¿no? También están mirando para acá porque los cambios que están ocurriendo actualmente en Laguna del Maule, dados por la acción de un volcán que supuestamente está debajo de la laguna, que provoca gases, que provoca agregación de nutrientes a este lago, es importante. Entonces, además se sabe que el fondo de la laguna del Maule se está levantando por acumulación de gases y de residuos magmáticos debajo. Entonces, claro, también es un tema bastante interesante de estudio y posiblemente alguna vez me vaya a dar una vuelta allá a tomar muestras.
Sí, posiblemente. Creo que Alexander Galán tiene una pregunta.
Sí, hola. Hola Alex, buena presentación. Te prendo la luz porque estoy aquí como súper interesante. Me quedan dos preguntas respecto del trabajo y es, o sea, ¿estás seguro que inicialmente no había aporte del glaciar hacia la laguna, no la negra, sino la otra, la que está más profunda? Es decir, realmente ¿tienes conciencia de que hay cambios en el aporte químico que tiene el sistema debido a esta interrupción del flujo que existía anteriormente? ¿O simplemente siempre ha existido los dos flujos y obviamente siempre hay un aporte importante de nutrientes? Y lo otro, bueno, dale, responde y te hago la otra pregunta.
Sí, hay un aporte de nutrientes desde el glacial hacia las dos lagunas. Siempre ha existido. Lo que pasa es que el glacial, al retirarse, disminuyó mucho, yo diría en más de un 80%, la cantidad de elementos que aporta a la Laguna Negra, porque al retirarse quedó orientado principalmente hacia la cuenca de Laguna Lo Encañado. Entonces, esto provocó una baja del influjo de nutrientes a esta laguna. Y esto significó el cambio en la laguna, además. Y en segundo lugar, el hecho de que estos pequeños lagunitas de retención, que no son tan pequeños, uno también podría cruzarlos en bote, ¿no? Genera un efecto en el que los nutrientes se quedan en estas lagunas de retención y el agua más clara va entrando a la laguna Maule, o sea, es una especie de filtro. Y eso es lo que cambió en estos años. O sea, primero, la entrada o la cantidad del flujo proveniente del derretimiento del glacial, porque digamos, el glacial ya se derritió tanto que ya no aporta tanto hacia Laguna Negra. Y además, el aporte que tiene ahora de sedimento quedan atrapados en estas pequeñas lagunas de retención.
O sea, la duda que me queda es, porque claro, hacen el estudio en este momento, ¿no? Entonces el estudio en este momento y ves la comunidad microbiana y bueno, todos los aspectos de la parte biótica, ¿no? Nutrientes, partículas, y ves que hay una variación importante, pero ¿cómo sabes que eso antes no ocurría? Si en el tiempo, si para atrás no había ese tipo de estudios. Es decir, ¿cómo sabes que eso realmente es el resultado del cambio en el retroceso del glaciar y en la interconexión hidrológica regular que había?
Primero, las dos lagunas también estaban unidas y tenían un flujo importante. Era un río que se formaba desde Laguna Negra a Laguna Encañado. Eso hacía que Laguna Negra aportara toda su microbiología, digamos, a Laguna Lo Encañado. No existen estudios anteriores de la microbiología, pero se pueden tener supuestos de que la microbiología era más o menos homogénea entre las dos lagunas, dado este efecto. O sea, una de las hipótesis que podemos decir aquí es que el hecho de que se hayan separado estos dos lagos provocó una diferenciación microbiológica. No lo podemos probar al 100%, pero es una de las explicaciones que nosotros proponemos al efecto que observamos.
La otra pregunta que tengo, golpearon una puerta. La otra pregunta que tengo es, ¿por qué dices que si la fotosíntesis es anoxigénica, porque los grupos que predominan aparentemente están bajo este metabolismo, o sea, no producen oxígeno, pero igual fijan carbono, porque la fijación del carbono va a ser igual independiente si es oxigénica o no oxigénica, ¿o no?
Claro. Claro, lo que pasa es que la anoxigénica lo que hace es utilizar sulfuro de hidrógeno o el hidrógeno como donante de electrones. Entonces, el tipo de clorofila es distinto y no fue detectado aquí en los análisis. El tipo de clorofila se detectó clorofila A, que probablemente provenía desde algas, microalgas o macroalgas que estaban presentes en el lugar, pero el aporte bacteriano en cuanto a clorofila, lo que teníamos ahí es bacterioclorofila. Y los metabolismos que se ve por la cantidad de microorganismos que existían son principalmente anoxigénicos.
Claro, sí, no, eso lo entiendo, pero al final en términos de balance de carbono, es decir, más allá de que produzcan oxígeno o no produzcan oxígeno, y lo que tú dices es cierto, ¿no? Claro, ahí el sustrato con el que reacciona el CO2 cambia, ¿no? Ya no es agua, sino es uno reducido como el H2S, por ejemplo. Y claro, no vas a producir oxígeno al final, sino un compuesto azufrado relacionado con el sustrato, pero al final la fijación de carbono es la misma, es decir, más allá es decir, que las tasas cambien entre una anoxigénica y una oxigénica, eso sería, obviamente, influiría en cuánto carbono estás fijando, pero el proceso como tal al final, es decir, no cambia que sea anoxigénico que no use CO2, porque me pareció que entendí eso.
Sí,
que no
sí, en algún
fijaban o
momento
que
me
disminuía la
pareció.
fijación, que de pronto esa aproximación es como, o sea, te genera como ruido porque al final eso no va a pasar.
De todas maneras, la anoxigénica es menos eficiente que la oxigénica.
Sí, definitivamente sí. O sea, puede
Entonces
que
la fijación
eso
de
sí tiene
CO2
sentido.
disminuye, sí.
Ahora, ¿cómo llegaste me perdí un poco, cómo llegaste a la conclusión porque las comunidades te indican que son más de tipo anoxigénico, por eso concluyes que es más anoxigénica la fotosíntesis, bueno, sí, la fotosíntesis, porque es síntesis finalmente con energía lumínica, ¿no? Que se transforma en química.
Fue buscando todos los microorganismos que fueran capaces de generar fotosíntesis. Dentro del listado de la base de datos se hizo una especie de búsqueda por base de datos, buscando los microorganismos capaces de producir fotosíntesis o de utilizar la luz o de ser productores primarios a base de luz, ¿no? Y el listado que me arrojó es el que estaba en el gráfico que mostré anteriormente, que son principalmente anoxigénicos y prácticamente, o muy pocos, que producen fotosíntesis normal, y esos están ubicados principalmente en la capa más alta de la laguna.
Y lo otro, ¿hiciste perfiles de oxígeno?
No. No.
Eso sería interesante ver cómo es la dinámica del oxígeno para ver si esas masas, o sea, ver la mezcla que tiene, porque qué profundidad tienen esos sistemas, ¿no son tan profundos o sí?
Sí, sí, son profundos, son 2000.
Eso
Déjame
sería
y te
súper
digo los valores
sería
exactos.
súper interesante, Alex, ver el oxígeno, cómo se comporta, porque me imagino que abajo debe haber una circulación bastante lenta, si es que circula, y deben haber procesos de anoxia asociados a los fondos de esto, sería ver todos estos procesos que ocurren en anoxia, cómo están operando en el sistema, ¿no? Y sacar muestras del fondo, súper interesante. ¿Vas a
Laguna Negra
volver?
tiene 276 metros de profundidad y Lo Encañado tiene 45 metros de profundidad. Las dos son bastante profundas.
¿Proyectos para volver tenemos?
No por el momento. No por el momento.
Bueno.
El proyecto Planetary Lake Lander ya se acabó. Están fijándose en otros objetivos. Una de las tentativas era la Laguna del Maule. Como bien dijo Aparna, es un tema bastante interesante. Otro también tenía que ver con las lagunas de la Patagonia, pero hasta ahora no se sabe si van a hacer continuidad en otros lugares, la ciencia cierta.
Bueno, ahí tenemos sustrato para tirar cosas y ir a buscar fondos porque está súper interesante. Súper, Alex, gracias.
A ti.
Alex, hay dos más preguntas también en YouTube. Uno de, no sé, de Daniel Arios. Esta creo que ahora eliminado, pero su pregunta fue que ¿cuál es tu sugerencia para muestrar en futuro?
En la misma laguna, podríamos hacer una mayor cantidad de muestreos en distintas partes de la laguna, porque todo este muestreo fue, bueno, por un tema de fondos también, en un solo lugar, en un solo punto central de la laguna, en distintas profundidades. Entonces, esto hay que ver cómo funciona el sistema más cerca de las orillas, en donde la profundidad es menor, la oxigenación es mayor, cómo funciona más al centro de la laguna y cómo funciona quizás en profundidades un poco mayores. Los 20 metros se eligieron porque era la profundidad hasta donde más o menos llega bien la luz, ¿no? Pero se podrían haber tomado en consideración más profundidades y más puntos de muestreo.
Ok. Ella también preguntando, ¿hay algún otro parámetro físico-químico o algo que considerarías para futuros muestreos?
Los parámetros físico-químicos, bueno, ahora que ya tenemos idea de cuáles son los parámetros que cambian, me enfocaría quizá en analizar más profundamente los parámetros que ya tenemos vistos. Ahora, como dijo Alexander, el oxígeno es un tema bastante interesante. La materia orgánica disuelta también. Podemos estudiar también otros metales pesados, por ejemplo, el efecto de algunos metales que puedan ser no buenos para los microorganismos o que puedan causar pérdidas, no como nutrientes, sino que todo lo contrario. Aquí una de las cosas que se vio es el arsénico, que estaba en muy baja cantidad, pero hay otros metales que también se pueden analizar. Aparte de eso, yo creo que los parámetros que elegimos fueron bastante, o el abanico de parámetros elegidos fue bastante amplio. Era un análisis exploratorio bastante grande, entonces no sé si se podrá analizar mucho más de esto. Ahora, como te decía recién, los puntos de muestreo para mí son interesantes. Hay muestrar distintas cosas o hacer un diseño experimental para ver cómo se mueven las comunidades perturbando el sistema. También es un tipo de análisis que me gustaría hacer.
Bueno. Y última pregunta de Almendra Espinosa. Muy interesante, gracias. ¿Cuánto tiempo tarda llevar a cabo una investigación como esta?
Esta investigación requirió 2 años. El primer año hicimos un premuestreo, hicimos mediciones de algunos parámetros básicos, hicimos análisis microbiano, vimos más o menos cómo se conformaban las comunidades sin hacer secuenciación masiva, sino que las estudiamos primero para ver la cantidad de muestras que teníamos que sacar, por ejemplo. Al otro año fuimos con todos los equipos, fuimos con un filtrador tangencial, sacamos 100 litros de agua de cada pozo. En realidad fueron 200 porque tuvimos que compartir también algunos de nuestras muestras. Entonces estuvimos 2 semanas en la cordillera acampando, en un campamento en donde teníamos también unos domos, laboratorios para poder trabajar allá mismo. Se hizo toda la etapa de prefiltración de muestras en ese momento allá en la cordillera y después los primeros pasos de la extracción de DNA también se hicieron allá. Se dejó en buffer de lisis y se llevó a los laboratorios de la ciudad para terminar de extraer para mandar a secuenciar. Entonces, en términos prácticos, para tomar 8 muestras y analizarlas, nos tomó más o menos un día por muestra, porque además teníamos que salir en el bote, teníamos que ubicar el lugar, tirar la sonda en el mismo lugar todos los días, y teníamos poco tiempo de luz, teníamos días con tormenta, y bueno, fue complicado, pero muy entretenido.
Bueno, gracias Alex. Y finalmente para continuar, por pregunta de Alexander, hay más lagunas en Maule, Lagunas Las Animas, Laguna del Medano, y todo en rutas de aguas termales. Entonces hay muchas lagunas en Maule para explorar y todo está dentro de área geotérmica de Laguna del Maule. Entonces hay más lagunas y creo que no hay trabajo también en estas lagunas, aparte del Laguna del Maule, porque el Laguna del Maule también está considerado dentro de Observatory Nasalovi. Pero otros no. Bueno, gracias Alex para tu excelente presentación. So today we are going to end up our webinar, the first day, 21st September of Webinar Internacional, the story of microorganisms in the context of International Microorganism Day. We had initially the talk of Dr. Deora and Dr. Angshu, who talked on different aspects of infection immunology on Bordetella pertussis, and then Dr. Angshu talked on antibiotic resistance and generation of knowledges. And then there was talk on extremophilic microbiology, and also Dr. Felipe's talk on using the industrial perspective of microbiology using the residues. So tomorrow at 9 a.m. Chilean time, which is GMT -3, we will again meet from 9 to 1:15 Chilean time. And tomorrow there will be 6 talks total by Dr. Joyce Hernandez from Mexico, followed by Dr. Kaushik from India, then Dr. Pedro from Chile, Dr. Alexander Galan from Chile, Dr. Celine from Chile, and finally Dr. Carlos Pedros from Spain. So see you all tomorrow, and see you all tomorrow at 9 a.m. Chilean time. Have a good day all. Thank you so much.